Hello.
Oh, sorry about that.
All good. Yeah, you like froze for a second. I figured that's what it was.
Yeah, yeah.
All right, I'll lock the door.
All right, thank you.
All right, take care.
Thank you. Yeah, I don't know. I think yesterday they were cutting down a tree outside, and it probably just messed everything up for everybody. Because right after I got cut off, I had like these Verizon guys knock on my door. So, okay.
Oh.
Yeah. So, all right. I just got back. Okay. So, yeah. All right. Wait. Let's
Yeah,
go.
so
You can
I don't
share
know.
it. Yeah.
I looked through the chem. So I feel like that's the section I was able to like kind of review the most. And the chem passages for the most part I didn't find as tricky. I guess the bio passages are something I really want to go over because
Yeah.
I really like don't even know where to start with them.
Okay. Yeah. Yeah. All right. You can share your screen
Okay.
again. And,
Yeah, is that what you usually do, like the share screen, or does that not really?
if it's something that I don't, because I don't think I did the sample test.
Okay.
But if it's something that I have done, then I could just have it open on my screen.
Yeah, but like in the future, so I just would send you the stuff beforehand. Is that how it like works?
Oh yeah, we could do it like that also, because that also lets me like get a head start on making like writing down explanations in the notebook that I'm going to share with you. And
Awesome.
if it still doesn't make sense after that, we can go over it. Yeah. So for the BB section, there's definitely, I mean, the passages are definitely going to be tough, but just go at them question at a time, like one question at a time, because an answer and like look at the passage if you need to, rather than trying to understand every little aspect of the passage first, because
Yeah, I think that's like my issue. I feel like that's what I'm doing. And then even,
yeah.
I mean, it's probably like this, like just seeing something like this
Oh
is
yeah,
like,
yeah,
well,
yeah. It's gonna be full of like real like a ton of like acronyms and things like that are just going to like be designed to like psych you out, because pre-meds are very like thorough, and pre-meds like to like read everything as if it's a textbook, and take notes on every little nook and cranny. But these are all like, if you scroll down to the bottom of the passage, see the source of it. They're all from basically published like research
mhm.
publications, which means that it's meant to be very like hyper specific and like obscure and niche. So you don't, yeah. So don't think of it as like you're reading up like something from a textbook and you need to know everything about it. They just want to be able to see if you can go through something like this type of literature and separate like, I guess, like the wheat from the chaff, like basically separate what's relevant from what isn't. But just remember that each passage just has like 5 questions, and most of the time, and if you just think of it as just 5 questions instead of the entire passage, most of the time, a lot of the things that look really like scary won't even be, they won't even ask you about it.
Yeah, okay. I mean, also
Yeah.
half the reason like this section was harder for me is because I don't have all of the amino acids like fully memorized right now, and I feel like that's like half of the freaking.
Oh, yeah. So for the amino acids, all you really need to know is, all right. So it's better to have like a. It's so for amino acids, like it's really useless to have them all memorized. All you need to know
Oh,
is,
really?
yeah, because all you need to know are the ones that are acidic, the ones that are basic, and why they are the way that they are.
So like the aspartic acid, glutamic acid.
Yeah.
Uh,
So it's kind of like, the analogy I like to use is, if you were to memorize the alphabet, you could just memorize the vowels, because there's only 5 vowels, and anything that's not a vowel must be a consonant, right?
yeah. Wait, what?
so
Oh,
the,
well, yeah.
yeah.
I think I get it.
Yeah, that's the analogy that I use. So for the 20 amino acids, 2 are acidic, 3 are basic, and therefore anything that aren't those five are just going to be neutral.
Yeah, but you feel like I don't need to know the structure because I feel like the only ones I know are like the non-polar ones.
Certain structures like will come up, but you know those, like for instance, like what's an amino acid that has its amine group as a secondary amine?
Probably histidine. I don't know. Wait,
so like proline.
Hold on, let me pull up my.
So you're thinking in the right way in terms of like, yeah, there's a ring, but that's a ring that's on the side group. But proline is the only one that is connected to the amine, the N terminal. So yeah, don't worry too much about those as we like go through the questions.
But wouldn't histidine be secondary also? Because both of the nitrogens are connected to 2 carbons.
Yeah, but they're not. They're on the side chain. They're not connected to the N terminal of the amino acid itself.
But doesn't that like not have anything to do with if it's secondary?
Oh, so like the thing with proline is that it is connected to the. So you know how every amino acid.
Weird ring.
What's up? Yeah.
It's like the weird ring, yeah.
Yeah. So that's an example of like a non-basic, non-acidic amino acid that is useful to know because it plays a part in like turns, like proline turns in like beta pleated sheets and stuff like that. So like in some cases, or they could ask you a question like, in this enzyme's active site, let's say you replace the amino acid with alanine, and alanine is like it's just like a methyl group. Yeah. So that's basically it's small, right? So when you think of that, think okay, so if we're replacing an amino acid in the active site of an enzyme with alanine, it's going to make it it's going to reduce steric hindrance, right?
Mm-hmm.
So if you can, because if you just memorize the structure of every amino acid by rote memorization, you may not be able to answer some of these questions. But if you think of them in terms of okay, alanine is small, so smaller steric hindrance, right? Proline is a ring amongst itself, so it's going to be playing a role in proline turns or things like that. But there's a video where it goes through sorry.
And stuff.
What's up?
I know little things about some of them, like the prolines, the helix, alpha helix breaker, and stuff like that. But then I can't tell you what tryptophan looks like. So
yeah, and you don't need to know.
I know it's a ring.
Yeah, it's gonna have whatever. It'll have 2 rings, but yeah, you don't need to know exactly what it looks like. Because you can, as long as you can make certain deductions, for instance, rings mean that they're pretty much non-polar, for the most part. So if they ask you which one of these amino acids is likely to, or which type of molecule can go past the lipid membrane, it'll be whichever one is non-polar, right?
Yeah.
So yeah, there's a video on my channel where I go through where one of my students was asking me the same question about amino acids, and we go through every single amino acid question in one of the section banks, and I basically show her how she can answer them without necessarily having to know every single amino acid, because
I think sometimes it's easier because I can if I see 3 of them that are different than the other one, then I know it's that one.
Exactly. That's how it is most of the time. But yeah, we'll see as we go through them. So for instance, well, it seems like you got this one, but do you understand exactly why?
Hydrolysis, yeah. I mean, that doesn't seem to complex.
You know what's meant by that? The gamma. Okay.
Wait, oh, sorry. The gamma
Yeah, the
phosphate.
gamma phosphate.
Yeah, it's like alpha, beta, gamma.
So it's the one that's furthest out there, right?
The third, yeah.
Do you know how kinases work?
Yeah, they remove the no, that they
Mm-hmm.
add the phosphate.
Well, yes, but they transfer it mostly. They're an example of a transferase. So they
Mhm.
transfer the gamma phosphate, right? Um, and they work on the amino acids STY, so
Oh, I didn't know
serine, threonine, and tyrosine. What do they all have in common?
that. You're gonna have to give me a minute.
Well, just think of any one of them.
Oh, wait. You said serine,
Mm-hmm.
threonine, and tyrosine. Oh, they all have alcohol.
Yes, good. And an
Okay.
OH group is basically something that is a good nucleophile, right?
Yeah.
Yeah. So those are the ones that kinases work on because they basically nucleophilically attack that gamma phosphate group.
Yeah, I remember that. That's like in every reaction in biochem, I feel
Yeah.
like.
Yeah. So that that's another example of like amino acids to know, but like go at it like don't go at anything like with rote memorization, but like always go at things with like associations. So like associate like STY with kinases.
Yeah, I feel like that's where I'm going wrong with my studying is like I'm like trying to
Mm-hmm.
bog down in like the memorization of the
Yeah,
smallest details in this
yeah,
book.
yeah. People spend an inordinate amount of time on content review without any
Yeah,
target in sight,
that's
so they
why
just kind of like you know they just feel like they're perpetually in this state of not knowing enough. But the best way to go about it is just like going through passages and answering questions because it's a critical thinking test, and the things that you need to learn, like you figure out what you need to learn, like you figure out what content to go over based on the questions.
yeah, so then um do you feel like then it's a reasonable amount of time for me? Like when do you think I should have my content review done? And do you think like
Oh,
I'm gonna
I
need to push my test back? Because I hear like
no,
kind of mixed reviews.
I think the to like just like don't even think of it as content review. Like basically the best thing for you to do is to start off with right now with the sample test and see what kind of material you need to know. Like we're gonna do that together like right now, and after that section banks.
Oh, so you're saying don't even read
What yeah.
these Kaplan books.
Yeah, I don't think you. Yeah, I because it's just it's passive, and you're not going to know like what you need to know and what you don't need to know. It's just going to be like this kind of like, I mean, you've taken the classes already, right?
So I'm like in the middle. I have them on Mondays and
Oh no,
Wednesdays.
I meant you've taken the classes like in college.
Oh, yes, I think for everything except for psych. Like I didn't learn any of
Oh
the
yeah,
psych.
yeah. So with psych, that's gonna be like I suppose the content review like section like that's gonna be like where you actually have to learn the stuff, but that's not that it's not that difficult aside from that
Yeah.
because they don't really test your critical thinking skills too much in terms of
Yeah, like I
that.
mean, I was able to do better on
Oh
psych,
yeah,
which
yeah, I saw that.
than like the other stuff, and I
Mm-hmm.
literally didn't even know it, but
Yeah,
just some
yeah.
of the words I can kind of like tell what it
Yep.
means.
Yep. So, all right, so let's just like go right at it. So, this passage, I mean, it seems like you're doing pretty well on this. Like, let's see.
Yeah, this passage I didn't have too much trouble with because I felt, well, I
Yeah.
don't know.
Mm-hmm.
Um, that one was way easy. This one was easy for me too
Mm-hmm.
because like I know like what enzymes do. Yeah, I don't really know if I knew this one at all.
Okay,
So
so for this one, right? So like, I would say go to attack the questions first. So I'm looking at the question. Proteins that are encapsulated in ARF1 COP derived vesicles are bound for this. And now I'm going to look for ARF1 COP, right?
Okay.
So, okay. It seems like, all right, so that's ADP ribosylation factor 1, all right. It plays an essential role in vesicle formation, okay. Responsible for the recruitment of cytosolic coat protein complexes. That's what COPs are. Wait, what?
Where is it? Oh, here?
Yeah.
Oh.
Mm-hmm.
Okay.
Yeah. So, okay. So then, and subsequent retrograde transport from the Golgi apparatus. Okay. It says localization to phospholipid bilayers.
Where does it say that?
Let's see. So, okay, proteins that are encapsulated in these vesicles. So, wait. Let's see. So, ARF1 is activated by these, nucleotide exchange factors, which replace the G part with a T. Or, wait, sorry. A GDP with a GTP, okay? And then when that happens, ARF1 changes shape, which releases this thing from a structural groove in the protein, okay? Localization to phospholipid bilayer.
I feel like this doesn't help at all with. I feel like this doesn't give me any information about like where it's bound to though.
Mm-hmm.
And it's not like anywhere down here. Like I had no idea what in here would be helpful.
Let's see. Let me see that top. The top. Okay. I wonder if it has anything to do with the retrograde transport.
Yeah, I have no clue.
Mm-hmm.
We can look.
Yeah, I think it's the retrograde transport aspect of it.
So how does that? What does it say? Retrograde transport from Golgi apparatus. So what does that even mean, though?
So so generally what happens is, all right, so like when some type of protein is made, right? Mm-hmm. There's like a post-translational modification, like from the signal peptide, which makes it go into the rough endoplasmic reticulum, and then stuff gets added to it or whatever, right? And then it gets sent to the Golgi, and the Golgi apparatus basically decides, is it going to be a cytosolic protein or like a membrane protein?
So after protein's made, the first place it goes, you're saying, is the rough ER? I thought the ribosomes...
Sometimes. Like, the protein will only... So, basically, like, in the cytosol, a protein will be, like it'll... mRNA will be translated into a protein, right?
Yeah.
All right. Now, if it has something called a signal...
yeah, I remember learning about that.
Yeah. It's just like a specific sequence that indicates that it needs to go back to the rough ER to be modified.
Wait, I thought the rough ER is where the ribosomes were, and that's where like the proteins were synthesized.
So that's where some ribosomes are, but there's tons of ribosomes all throughout the cytosol, and that's where most of the proteins get made, right? Yeah, it's only with the signal, whatever. I forgot what it's called. You know what I mean, though, right?
Yeah.
It's only that indicates that it needs to go into the rough ER.
Okay.
To get modified. And then after it gets modified, it gets shuttled to the Golgi. And then the Golgi sends it either to the cytosol or to the membrane. So it seems like it eventually goes to the membrane.
So then retrograde transport would just mean that it's like going backwards. It would just be going
Yeah.
back to the ER.
Mm-hmm.
Okay. So
Yeah,
I
this
guess
one could have been tricky though because it eventually does go to like it says localization in the phospholipid bilayers, right?
yeah, I don't know. I put the cytosol. I didn't really know my. So I took this. Let me just like preface. I took this in May.
Mm-hmm.
I took this the end of May.
Yeah.
So, I mean, I've learned a little bit since then. Not
Mm-hmm.
too much, but
Mm-hmm.
I gotta remember any of these questions or like why I picked anything.
Yeah. So yeah, basically what the answer is because of the retrograde transport from the Golgi.
Yeah. Okay. So I just didn't really know what that. I think
And
when I saw like the protein stuff, I just thought
mhm.
like cytosol.
Yeah, and then the first paragraph, if you yeah, that's talking about biomolecular trafficking, right?
Yeah.
Meaning like movement and stuff. Oh, and so that means including protein packaging by the Golgi apparatus. You know how they say that's like the mailbox or whatever, or the post office. So that's what so all right. So that's where the protein packaging happens where it gets sent to different places, right?
Yeah, I think my thing was I know that that's like the post office, but I just didn't know where it's sending it and what sending it to.
Yeah. So it'll send it either to someplace in the cytosol, like most proteins, or like it'll become like a structural protein in the
In the membrane.
yeah.
Yeah. Got it.
So
All right.
but the keyword here is the retrograde transport from the Golgi.
So would you just not like I just want to make sure I get how you're approaching these. You wouldn't go through you'd like skim the passage first? Is that what you're saying?
I would look at the question first,
Okay.
and then depending on how difficult the question is, I would read as like whatever I need to read.
Yeah, because I feel like for the
Because then you're
first
looking for
three.
specific things, right? Like I see the ARF cop thing, so I look for ARF cop.
Okay.
Because if you
Yeah.
just read it without the questions, you don't know what to focus on. Am I going to focus on BFA? Am I going to focus on fungi? Am I going to focus on AMF 26? Right?
Time. Yeah, I
Yeah.
get
There's
that.
just so much going on, and like this is like this is a literal like research paper, like AKA like something that has before 2012 wasn't discovered. So it's not supposed to be like easy like a textbook or anything like that.
Yeah, no, that makes sense. Because like I don't know if it's just easier for whatever, but like these I definitely didn't like I didn't need to read this to know what kind of
Yeah.
enzyme that is. So
Yeah.
like that does make sense and that probably would save me a lot of time because I know for
Yeah.
this I mean, I was distracted because I took it at home, but like I was definitely running out of time. I was also just trying to comprehend all of this before I answered.
Yeah, you don't even have to. You don't have to comprehend every aspect of it. Just answer, deal with it as much as you need to in order to answer the questions. There's
Yeah,
only
like this is
5
where, like,
questions.
crazy highlighting everything.
Yeah, I see a lot more of that here.
I think I was probably a lot more confused on this one.
Yeah, so like, I would highlight stuff like mostly like nouns. Like I would just highlight like ischemia or however you pronounce that. And then I guess loss of blood flow, and then
So I
it's
remember
bad.
like looking at this question, I definitely had so much trouble with this one.
Okay, so all right, best supporting evidence that neutrophils are the cause of the reperfusion injury. So first thing I'm gonna do is look for reperfusion injury. And after a while, like you'll get used to being able to just find like to do like a control F thing in your head.
Okay.
So, so yeah. So I'm gonna look for the whatever the
I also like.
Yeah.
Am I supposed to know what reperfusion injury is? Like, I have no clue what that is.
Oh no,
Okay.
because that's just going to be in the passage, right?
Okay.
We're basically gonna look for reperfusion injury, and we're gonna see which choice, if it were true, would provide evidence that the cause of reperfusion injury are neutrophils.
Okay.
So first, let's look for reperfusion injury.
Yeah. I'm trying. During reperfusion. Okay, I don't see them together at all, but I see like injury stuff here and reperfusion here. I could have missed it though.
Mm-hmm. Okay. All right. So I see in the first sentence.
Reperfusion again, yeah.
Yeah, and tissue injury. So it seems like they tell you that ischemia is a loss of blood flow. And then it says subsequent blood reperfusion, which makes me think that that means that it's the return of blood flow.
Okay, yeah, that makes sense.
And the first thing that says tissue injury means that as that reperfusion occurs, which is the return of blood flow to the tissue, there could be some type of injury involved to the tissue.
I feel like I would just never be able to
Mm-hmm.
put that together.
Let's see. The
I
tissue
get
injury.
the
Yeah.
reperfusion. I get where you're going with that.
Mm-hmm.
So loss of blood flow, then after that you're putting blood back in.
Yeah, because you're saying the tissue injury that results from ischemia and subsequent blood reperfusion.
Oh, okay, yeah.
So and subsequent means like after, right?
Yeah, right after.
So it's not talking about the tissue injury that results from loss of blood flow, but it's talking about tissue injury that results from loss of blood flow that's followed by the return of blood flow.
Okay.
So
that makes sense. And then should I keep looking in the passage then?
So then I see something that says until recently they thought that tissue damage was just from anoxia, the lack of oxygen.
Where are you looking?
Oh, the so okay, I'll just say how I would go through, yeah, cuz that first paragraph, what I pay attention to is what I would highlight is tissue injury, ischemia, subsequent reperfusion. And then I would highlight maybe, or at least pay attention, I'm not going to pay attention to underlies several common life threatening disorders including blah 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 blah.
Yeah.
Because I get it that there's tissue injury and it's bad, right?
Yeah, no, that makes sense.
Yeah, and then at the end it says that until recently they thought that the tissue damage was just caused from anoxia, which is a lack of oxygen. And I want you to tell me, what is the connection between ischemia and anoxia?
In general or from this article?
In general, which you can figure out from this article.
Okay. Lack of oxygen. They're saying that they thought the tissue damage was just from the lack of oxygen.
Yeah. Which is... So, I mean, what's the point of blood?
To carry everything in our body and...
Everything?
No. Bicarbonate. Oxygen. I don't know.
Yes. Oxygen,
Okay.
yeah.
Oh yeah, like the hemoglobin
Mm-hmm.
with oxygen.
Yeah.
Okay.
So yeah, tell me what's the point of blood?
To carry oxygen to our tissues.
Yes, good. So if we have a lack of blood flow, right, there should be a lack of oxygen, right?
Oh yeah.
Because that's what
Makes
carries the blood.
Yeah, so it's saying until recently it was believed that the tissue damage was lack of oxygen.
Which I
Oh,
would just say equals the same thing as ischemia.
okay, that makes sense to me.
So they're saying until recently, right? We used to think that the tissue damage was just caused by ischemia.
Okay.
But since they said until recently, right? There must be something with the reperfusion that is also causing that tissue damage, right?
Wait, I thought so. They used to just think it was from no oxygen, but now they realize that it's from no blood flow. And
Well, lack of oxygen and
oh,
loss
it's
of
the
blood
same thing.
flow are equal, I would say.
Oh, so but we would have to put that together.
Oh, well, yeah.
Yeah. Okay. I definitely did not put that together.
Yep, but you see, like, now how they're the same.
Yeah.
So, like, I would, um, like, in, like, notes or whatever, like, I would write ischemia equals anoxia.
Wait, in notes like on the MCAT?
Oh, no, just, like, in, like, stuff that I would write down. Here, let me see if
Oh,
I could do this. Um, Here, I'm gonna share my screen for a sec. So, here.
so I have to stop probably.
Oh, no. I have it so multiple people can share.
Oh,
So,
should I read it then?
oh, yeah. But here, so I would just write this. So, tissue damage equals ischemia plus reperfusion. Right? Do you see what I'm writing?
Yeah.
And then I would put this, like, equal sign between ischemia and anoxia.
So would you write this like before you look at the questions? I guess I'm trying to figure out like how, or we're just doing this now that we saw that we have a question on this.
Yeah. Well, in terms of, like, understanding a passage, uh I do like to write things down in, like, a form of, like, shorthand for me to understand what the passage is saying.
Yeah, no, that makes sense for sure.
So, like, basically that paragraph I could sum up by saying tissue damage equals ischemia plus reperfusion, right?
Yeah.
And I'm equating ischemia with anoxia.
Yeah. No, I get that now. I
Mm-hmm.
during the test I definitely didn't.
Mm-hmm.
But I mean that makes sense, like now that we went through it slower.
Yeah. So, now what we're going to really focus on is the connection between tissue damage and reperfusion.
Okay, so do you think that that's enough? So then after
Oh,
that,
so that's just for that first paragraph. Now I have to make the connection with neutrophils.
okay. Oh, okay. So now we gotta look for neutrophil
Mm-hmm.
is
Yeah, so
right.
they basically found. Okay,
I don't know the
so during
leukocyte
reperfusion, neutrophils release toxic products.
during reperfusion.
Do you see that where it says that?
Where
It's it's like in the
getting
middle of the second paragraph. You didn't. It's like, yeah, like to the left of
up?
that. See where it says during reperfusion?
Yeah. So you know, it doesn't really matter that. Like, would you just kind of ignore that they adhere to the? I guess I have a lot of trouble figuring out like what's important.
Yeah. So yeah, that's one of the things I like. I guess I specialize in. So I see that it says during reperfusion, neutrophils release toxic products, and then the scientists hypothesized that most of the tissue damage was actually a neutrophil-related phenomenon. So, I'm sharing this again. So, see where it says reperfusion? I drew an arrow saying in neutrophils, toxic products are released, and then I put this equal sign, meaning toxic products equal tissue damage.
Wait, hold on, I can't see. I have to like unshare mine to see.
Okay, yeah.
Okay, so all right, so you do this. Tissue damage equals reperfusion. I feel like I'm gonna have time to do all this though.
Well, think of it as like something that initially takes time to learn how to do that will save you like an exponential amount of time as you do it. Because as you do this, like it's like you start to like internalize it and be able to do it in your head.
Yeah, so I think like I tried to do maybe it was for cars, but I think I tried to do like shorthand notes, and then it was like I don't know if this is what I'm supposed to be doing, so I just.
Oh yeah, I do like shorthand notes like all the time. Like here, for instance, like here, this is like kind of like some tutoring for like some students of mine. So like let me see what I can show you. Like, let me see. Wait. Like this was like. All right, so here, this is like kind of like the type of stuff that I write. Like I like to use the proportional symbol.
Yeah.
And I like to use arrows, and if it's inversely proportional, it's like that. So this just basically helps me equate things because there's just so many. There's just so many like acronyms and an excessive amount of terms.
Especially for physics. Oh.
Well,
that helps.
Physics surprisingly isn't like too verbose, but we'll see it together. So okay, back to that question. So you can share your thing again.
Okay, let me find it. Sorry, I have like so many tabs open. This is not what I need. Here it is. Oh, I gotta escape full screen. Okay.
Okay.
So then we got their toxic products, and we don't care that they adhere to this.
Not really, right?
Okay.
They're just asking us which of the following experiments would provide the best supporting evidence that neutrophils are the cause of reperfusion injury, right?
So, oh yeah, because we're not reading this until we look at the first question.
Yeah, pretty much.
Okay, cool.
Okay, let's see. Performing the ischemia reperfusion experiment using animals whose B cells are depleted. So we don't care about B cells, right? We're talking about neutrophils.
Yeah, I didn't know what that. I also don't know what B cells do. Well, it says antibody producing, so I guess.
Oh yeah.
I remember I read this first, so I tried to understand it first. That's why, if you look, I took an absurd amount of time for this question. And I think I saw something about I focused on the third paragraph for this question.
Oh, okay. I haven't even looked at that yet.
Yeah, so I did, and then I saw that the antibody B had the reduction. This is where I went to choose a different answer.
Mm-hmm.
And I was like, oh, well, antibody tissue injury.
Mm-hmm.
I don't know if I chose A. Oh, I chose D, so.
Yeah, even if some type of test did show that, they're asking just about neutrophils, right?
Yeah, so that's what I thought, but then it is about neutrophils, the experiment, I thought, but I could also be understanding the experiment really wrong.
Oh, okay. Well, they're saying neutrophil adhesion receptors, like they're describing the protein of the adhesion receptors for neutrophils, right?
Yeah.
Like basically neutrophils, right? They adhere, they stick to part of like the cell, right? And so they're describing the receptors of the cell as heterodimers with an beta subunit and an alpha subunit.
Yeah, which probably isn't that important, I'd say.
Well, because they're not on the neutrophil, right? They're just the thing that the neutrophil sticks to.
Okay.
Yeah.
That kind of,
And then they're saying that the subunits, they isolated them from neutrophils and then they used them to make antibodies. And so antibody A is going to be directed at the alpha subunit and B against the beta subunit. And then they say,
yeah.
I'm just going over this, like even though we don't have to. And then they say that only the ones that received antibody B showed a greater reduction in subsequent tissue injury. So that means that the beta subunit of the neutrophil adhesion receptor, that is the one that causes basically bad stuff to happen, like tissue injury.
Oh. Okay. Wait, that kind of makes sense actually now that you went through it. Like if this is B is the beta subunit, and then if the only ones that got the B had the reduction, so that means that like antibody A probably has no real effect on the tissue.
Mm-hmm.
Okay. So then why did I choose? Oh wait, so we don't care about the antibody B then?
Yeah, so basically they took the neutrophils, the neutrophil adhesion receptors, right? They separated the protein subunits, and they put them in mice to generate antibodies, and B cells are the ones that generate antibodies. Right?
Yeah.
But it doesn't really matter how we made the antibodies. We just are using them to see which particular subunit of the neutrophil adhesion receptor is related to the tissue damage.
Wait, oh, so this one would just tell you, like, okay, yeah, it's the beta subunit. This wouldn't tell you anything that neutrophils are the cause of it, right?
Oh, let's see. Performing this experiment using animals whose B cells are depleted. That just means that those animals are going to get tissue damage, and
So
we
that-
won't even know anything about the subunits because they don't even have the B cells, which make the antibodies.
Okay. I think I didn't choose B also because I have no idea what like this means.
Mm-hmm. Yeah, but I mean, if they're in A and B, we can kind of ignore it. But I guess like you're saying that. I guess you're saying that compared to C and D.
Yeah, I don't really know. I think I was just like thrown off by the fact that I have no idea what this is. I mean, I know this is like mentioned earlier, and so is this, but I didn't really know like what that experiment is or like if that's something I needed to know, and I just didn't know yet.
Okay, so like here, I would do this. Here, I'm going to.
Are you sharing your screen?
Yeah.
Okay.
Yeah, this time I made a screenshot, so we don't have to like keep going back and forth. But here, check this out. So all right. Basically, I'll explain like what each of these choices are saying. So this is saying, so okay, this is just saying no antibodies produced. Using, animals, examining whether it's reduced. No antibodies produced versus antibodies produced. Right? And then here, this is saying antibody against alpha and beta subunits.
Oh, that's pointed to C. Okay.
Oh yeah, sorry. Here.
Repeated.
And then let's see, another antibody against the beta. Okay, and this is beta only. So okay, A is saying we're gonna do the experiment, right, using animals who don't have B cells, right, and we're gonna see if the tissue damage is reduced, right.
Oh yeah, but that kind of like makes I don't know.
So we're basically comparing a group of mice that have no antibodies produced versus a group of mice that have antibodies produced.
Okay.
But or rather we're just really just looking at mice that have no antibodies produced. Because they don't have the B cells, right?
Oh yeah. Okay, that makes sense. If they don't have B cells, then they don't have any antibodies, so they'd have more tissue injuries, probably like what we'd see.
Yeah, so this isn't going to really change anything, right, regarding neutrophil caused tissue injury.
Well, I guess I'm confused because I feel like this makes sense to me now. I don't know. Because I thought like if it didn't have antibodies that it would change the neutrophil like tissue damage.
Yeah. Like, okay, so then all right, so let's look at C and D then. So
Okay.
C is involving an antibody that goes against the entire heterodimer. So it's an antibody that goes against the alpha and beta subunits.
Okay. Which
And then here, I'll just write neutrophil adhesion receptor equals alpha plus beta. But then alpha is the one that's related to tissue damage.
so the alpha is the one that causes the tissue damage, which we
Mm-hmm.
and I thought we said the beta was. Oh no, because the beta doesn't have any. I don't know why.
So in other words, antibodies against the alpha subunit
Oh yeah, yeah.
will
Okay, now
cause
I have okay.
will cause a reduction in tissue damage.
No, I thought that antibodies against the alpha had no effect, is what we found, what the passage says.
Oh crap! Is that what it said?
Yeah, okay. I think that's why I'm confused. Yeah. I can share the passage again.
Okay. So let's see. Antibodies against.
Yeah, because it says antibody A had no effect.
Got it. Okay, sorry. So antibodies against beta. Did they say yeah? Beta cause
Yeah,
a
okay,
reduction
now-
in tissue damage, right?
Now I get it, yeah.
So okay, that's what we're looking at. So look, if no antibodies are produced, like in situation A, it'll be no different. Like no difference, right?
If no antibodies are produced, then I thought we'd just have like a lot of tissue damage.
Yeah. So it'll be no difference from because all of the neutrophils are still going to be doing their damage, right?
Yes.
So we're not going to gain any information from that,
Okay.
right? And then if it's an antibody that's against both of the subunits, it should reduce the tissue damage. So this one would reduce tissue damage. I'm just going to abbreviate that as TD.
But I feel like it would reduce it by the same amount, no?
It would.
Oh, okay.
But so we'll come back to that one, and then for the beta only, it'll be a no difference as well. Now, if you look at B, they're doing it using animals that don't have neutrophils. So here,
Yeah, because D, I get why D is none, and I get why C is we decrease because you're adding another antibody. And then A, if there's no antibodies, wouldn't you? A would be increased, though, I thought.
So if there's no antibodies produced, then all the neutrophils that are causing the tissue damage are still going to be causing that tissue damage. So basically, we're trying to see what is the cause of the injury, right?
Yes, but I thought the antibodies are what like makes less tissue damage. So if you didn't have any antibodies, then it would increase.
So the antibody. So if there's no antibodies, right?
Mm.
Versus, so I guess I shouldn't have crossed it out, but let's say let's compare it to if the antibodies were produced.
Yeah.
Then there will be a difference. But all we're trying to just find is evidence that support the I guess hypothesis that neutrophils cause the injury.
Okay, so then we're looking at B now.
Yeah. So basically, if let's just say you have two groups of mice, right? And
Wait,
yeah.
wait, just reading B, that like makes a lot of sense.
Yeah, because if you have a group of mice so there's a control, right? Which means that there's no antibody situation happening. There's no it's just normal, right?
Yeah.
So let's have that control, and then let's have another group of mice that have no neutrophils, and compare the tissue damage between them.
Yeah, wait, that makes so much sense. I feel like
Mm-hmm.
I don't know. I was
Yeah.
really going through the answer choices, but just reading B now, I'm like, okay, wait, obviously.
Mm-hmm. Yeah. So basically, when they're talking about an antibody against alpha and beta subunits, like that is going to be just to the entire point of the entire point of the antibodies is to determine which subunit of neutrophils is the one, that's guilty, right?
Yeah.
Yeah. But since we're only trying to find evidence that support the hypothesis that neutrophils are the cause of the injury, the best evidence for that is comparing mice with neutrophils and mice without neutrophils.
Yeah, no, that makes like complete sense. Now that I'm just like reading it, I think I just like the words kind of just tripped me up.
Yeah, because the thing with C is it's it's still talking about um. So it may have the same result as reducing tissue damage because the antibodies fight the neutrophils, but you
Yeah.
still have neutrophils in that mice in the mice that have those antibodies.
Yeah, no, that makes sense. All right, I'm gonna run to the bathroom, but I'll share the next
Yeah.
question.
Here,
If
I'll stop
that's
my
okay.
share. Yeah.
Wait, hold on. Information is. Oh, actually, I did get this wrong. The not this question, but the question after this, I was more confused on. But I guess I still should go over probably this.
Okay. So they're saying information in the passage suggests most strongly that the function of the beta subunit involves. Okay. So that is the thing that is part of the adhesion receptor, right?
Yeah, and that's what the antibody was targeting. So that we figured, didn't we just determine that the B subunit is the one associated
Oh,
with
you
it?
know, by the way, I just noticed something from that previous question.
Oh yeah. Do you want me to,
C and D don't talk about performing the ischemia reperfusion experiment.
Yeah, but how did you know that? Like, I think why I chose D was I was like, oh, I should probably repeat the same experiment and change one thing.
Yeah, so they're basically just talking. Yeah, they're not telling us, since they're asking us evidence that neutrophil, like, find evidence that neutrophils cause the reperfusion injury. We have to do that reperfusion,
Oh,
ischemia. Yeah.
whoa. Okay.
Mm-hmm.
All right, that makes sense.
Yeah. So choice C would be possibly like an answer if they did something like, if they did the reperfusion ischemia experiment again. So I mean, sorry, not the top part, but that first part is what's important.
C would be an answer.
If it said performing the ischemia reperfusion experiment, yeah.
But I feel like this is, I don't know. But it doesn't it have like nothing to do with neutrophils though now?
What? Which part? Well, it's saying, if they said performing the ischemia reperfusion experiment using antibodies directed against the entire alpha beta heterodimer and examining whether the degree of tissue damage is reduced, that would be, that would be an answer, yeah.
Oh, interesting.
Because the antibodies would be basically not letting the neutrophil adhesion receptor like function, which means that it would produce a similar type of end result as not having neutrophils there at all.
Because it would go.
Because you see where it says in the passage, during reperfusion, adherent neutrophils release toxic products, not just regular neutrophils, but the ones that adhere.
Yeah.
So if you can basically get rid of that, if you can like have neutrophils that don't adhere, it shouldn't be causing any trouble.
Oh, okay. So I see what you're saying because the adhesion receptors are the heterodimer.
Mm-hmm.
So if you have antibodies at that whole heterodimer, then it wouldn't be able to adhere.
Yeah, and it says that It says that the adherence of neutrophils, like, that is what causes the release of toxic products and stuff.
Yeah, because I see that and I
Mm-hmm.
get that, but then you're saying, so obviously if you're attacking this, then it can't adhere, so there couldn't be any toxic products.
Yeah.
Okay. Yeah, I mean, it's tough because I get it now that like we're going over it slowly, but
Yeah.
it's hard. I feel like during,
Mm-hmm.
especially with counting down, it's.
Yeah. But it always starts like this. It's like we'll spend a lot of time on like these very like specific like the wording and the subtleties and the nuances of these types of problems, and you'll just like learn exponentially. So like
Okay.
by the time you take the test, you'll already have trained your brain
Yeah,
to be able to see what, like, how to like, I guess, organize like the thoughts and stuff.
I feel like so lost when I look at these, especially graphs.
Yeah.
Sometimes get me.
Okay, let's see. So, okay, what does a beta subunit do, right?
So that's one of the adhesion receptors, and
Yeah, so only the animals that received antibody B, which directs against the beta subunit, showed a great reduction in subsequent tissue injury, right?
Oh, so it's most likely that they like are involved in the adhering.
Yeah.
Oh.
Yeah, and the generation of the antibodies, that's done by the beta cells. I mean,
Oh,
not the beta cells, the B cells.
no, that makes sense to me now. But this one, I think I was like really confused on.
Let's see. A scientist claimed that antibody B offers a better means for preventing organ injury than agents such as free radical or protease inhibitors. Okay. Which of the following reasons offers the best support for this claim? Um, all right. So basically, they're saying that. Let's see. All right. So we know that when the neutrophil adheres, it releases oxygen-derived free radicals, proteases, and prostaglandins, right?
Oh yeah. Right here.
And so, okay, if we have antibody B, right, that means those things aren't produced at all, right?
During adherence, we're releasing all of that. So the scientist is claiming that that offers better means. Oh yeah, because if you just inhibit that, then it's already adhering.
Yeah, so not only does it so like that they're saying free radical or protease inhibitors, that means that we're still letting it make free radicals and proteases, but we just inhibit them after they're already made, and it doesn't say anything about prostaglandins either.
Yeah, no, I actually get this now because we're blocking like it adhering so that nothing's gonna be released. Yeah.
Yeah, it's better. Yeah, it's better to like stop it at the source before. It's better to stop something releasing toxic products than let something release toxic products and get rid of like two out of the three.
Yeah, no, that makes a lot of sense. I don't know why I chose C.
Antibody is very specific, therefore it will not recognize. Yeah, I guess because you're saying that. I guess you were thinking that of an antibody as attacking the toxic products when it's just attacking the adherence of the neutrophil, which causes like the toxic products to be released.
Yeah, well, I feel like I've just read this and I was like, oh yeah, specific. It'll just recognize the beta subunit. That must be right.
Mm-hmm.
But no, this makes way more sense now that I'm like looking at it.
Yeah.
Um. Okay.
Let's see. The scientists wanted to use antibody B clinically to treat humans. All right. Well, first of all, they're doing it in mice, but okay. But this proposal was rejected. Which of the following is the most logical reason? Yeah, because it was it was made in mice. So, well, I guess they all say that, so that's obvious. Let's see.
Yeah.
It can never be used in humans. Repeated usage in the same patient would elicit the production of human anti-mouse antibodies.
I'm so confused by
It will
that
not
one.
recognize human antigens. It can only. All right. So first of all, So
I know why
Yeah.
I chose D for this. I chose D because I was like, okay, well, if it's in mice, then you can't like you can't make it in your body. Like it's gonna need to come from somewhere else.
So,
Um,
do you know what in vitro means?
Isn't it like oh it's in your body, isn't it?
So that would be in vivo. In vitro is like in glass or like a test tube.
Wait, so yeah, I think that's why I picked that because
Mm-hmm.
if it's in mouse, then we'd have to do it in the test tube and then insert it into the humans. Is what I was like thinking.
I mean, like if it is made in a live animal, right?
Oh, then it's in vivo.
Yeah, you can use it in. It says it can only be used in vitro, but I mean it works in live mice, right? So that's an example of it being used in vivo.
Yeah, I think. I don't know. I just assumed that since it was in mice, like the humans wouldn't be able to do it, but I don't. Yeah.
Well, yeah, we're so okay. The fact that it's made in a mouse is definitely going to provide some type of like complication here, but we have to
Mhm.
see what complication, arises, right?
Yeah.
Now, we definitely know based on what they say, because they said when these antibodies were applied to lab animals subjected to ischemia of the heart, only the animals that received antibody B showed blah blah blah, right? So they're already doing in vivo experiments, right?
Yeah, when these antibodies were applied. Yeah, so it doesn't specifically say it, but yeah.
Yeah. So if they say it can only be used not in animals,
Oh.
that's not
Oh,
true, right?
yeah. Okay. Okay, I get that now. I think I X'd this out because I was like, what is a human anti-mouse antibody though?
All right. That's what I was gonna say about C. Human antigen. So what's an antigen?
Isn't that it? It's with the antibodies, no?
Yeah. What with it though?
Um, does the antibody bind to the antigen?
Yeah. But like what? Tell me more about the antigen.
I'm
Like,
not sure.
is the antigen, like, where does that come from?
I want to say like your immune system. I have no idea.
Well, the antibody comes from the immune system, yeah. The antigen is just the part of the pathogen that the antibody it recognizes and binds to.
Oh,
Yeah.
okay.
Mhm.
Like so it's on like what you want to kill.
Exactly. Yeah. And the antigen is like the, I don't know, like it's like, let's say like a virus or something. Let's say like COVID or something like goes into your body, God forbid. Um your body isn't going to recognize COVID, like, the coronavirus itself. Like, part of your body will like phago, like part of your immune system cells, like your leukocytes, like your neutrophils, basically. They're going to phagocytose, like they're gonna eat, like, the virus,
Yeah.
and they're going to present like a protein from the virus that is just specific for that virus, and that is what it's going to use to show the rest of your immune system, like, this is what we're looking for, this is our target, like, this is, like, kind of like if your immune system cells are like police officers and stuff, and they're like, okay, we're doing like a nationwide manhunt, this is like what, this is the what the person looks like, this is the image of, this is like the police sketch, that's the antigen, basically.
Okay, yeah, I think I get that.
So if they're talking about human antigens,
The antigen, but I don't think I like processed
mm-hmm.
that whole thing. So like it wouldn't be a human antigen, you're saying it'd be like a virus
Yeah,
antigen.
or like, yeah, something like whatever, yeah, like a pathogen of some sort, yeah.
So it in order to make this right, it would be human pathogens.
Oh, it just wouldn't be human. It would be, I mean, in this case, they're not really using it for pathogens, they're using it for uh the adhesion receptor,
Okay.
like the subunit, right?
Antibody B clinically.
Yeah. So it should say it will not recognize, let's say, like, maybe like human, like human neutrophil receptor, or something like that. It shouldn't say like it should specify the pathogen, or in this case, the subunit of the neutrophil,
Okay.
but
That makes
not human, because that's like saying that it's an antibody that's designed to kill anything that's human.
okay, yeah. That's not what we want.
Yeah.
But then what is a human anti-mouse antibody?
Okay, so all right, so this is a little harder now. So like if we look at A, I'm just going to look at A first for a second. They're saying that it can never be used in humans, right?
Yeah, I X that out right away because that just seems like a very bold statement.
Exactly. So I would say like same thing with D. They're saying absolute statements like never and only. And those statements tend to be like wrong because if they're talking about absolutes, it's very easy to disprove, right?
Yeah,
Because
that's what I did in the cars.
Yeah. All you need is one counterexample, right, to disprove an all or nothing type of statement, right?
Yeah.
So
Okay.
yeah, so I would steer clear of A because it says never. I would steer clear of D because it says only. Now B is the only one that says like it would elicit the production of, meaning that it would make some of this. It doesn't say that it'll make only this. It wouldn't say that, it won't ever make this, right? Okay, so now B is saying since the antibody was generated in the mouse, repeated usage in that same patient would elicit production of human anti-mouse antibodies. So maybe it's just that they're saying anti twice, but if you cross out the anti-mouse part and just look at human antibodies, it would do that because over time, like they say repeated usage in the same patient because it takes time for your immune system to make antibodies.
Okay.
Because and because they're specific and they're slow to make.
Okay, I don't.
So basically what's going to happen is that your body is going to be like is going to treat antibodies made by a mouse as a foreign like substance.
It's
Okay. So basically what B is basically saying is that over time your body is going to recognize that these antibodies that are made in a mouse are not yours, and therefore your immune system will view these mouse made antibodies as a foreign substance.
Oh. Yeah, I get it now.
So like some of the clues that I see are repeated usage, same patient, and then elicit the production of. Because your B cells make your antibodies, and that takes a long time in terms of like your immune system, right?
Yeah, so then if you had the human anti-mouse antibodies though,
Mm-hmm.
why would this... Oh, because... Like, why would we not be able to use this?
Oh, just because over time, right? A human immune system will be like, that is from not you.
Oh, so the antibodies would be killing this.
Yeah, we will make our own antibodies to kill that antibody, yeah.
Yeah, that makes sense.
Oh, okay. There's a new one. All right. A neutrophil has point mutations. All right. What's a point mutation?
Okay. Isn't that? I could be wrong. Isn't that just like when there's a single base pair change?
Correct.
Okay.
Now, yeah, what can happen? Like, what? So it can be a substitution. It can be what else?
It could be substitution, insertion, deletion.
Okay. What? So if there's an insertion or deletion, tell me how that can like really wreck, like, yeah.
Reading frames, like,
Good. Perfect. Yeah. It would mess up the reading frame, right? Unless it's in a multiple of 3. Okay, good.
Yeah.
So anyway, a neutrophil has point mutation in the genes coding for the alpha and the beta subunits of the adhesion receptor. However, this cell can still migrate through endothelium. So, which of the following conclusions about the effect of this mutation can be drawn? Okay, so I look up at the second paragraph, or rather the third paragraph. It says that neutrophils adhered to the vascular endothelium, right? And then migrated into surrounding tissues, right?
Oh, we're looking at
Yeah.
right here. Yeah.
Okay. So now they're saying that, so they're saying that some neutrophil has some type of mutation for those subunits, but it can still migrate. It can still do what it basically does, right?
Yeah. Okay, so it has a point, but it can still do what it's supposed to be doing.
Yeah. So looking at the other choices is what will help you here. Because it seems like it still works, right?
Okay. Yeah.
So it should still be able to release prostaglandins, right?
Uh-huh.
I mean, it's beta subunits, which is the thing. Yeah.
Wait, so also could be like exocet because it's only...
Yeah.
Okay. And also the beta subunits don't really... Oh, but I thought...
Yeah, so the beta subunits are the ones that cause, whatever the tissue injury.
Oh, but it doesn't...
But we don't know anything about the alpha one. Because basically, if you get rid of the alpha, there's no effect on it, right?
Oh, wait. So this actually makes so much sense now, because if it can migrate through, then it can probably still bind.
Yep.
Okay, yeah.
And since the alpha subunit basically has no effect, like if you get rid of the alpha subunit, it will have no effect, like it'll still be able to bind. We don't know if it has an alpha subunit or not. So we can't say that it only has the beta one.
Oh, I see what you're saying. Yeah.
Yeah. So it's
Then-
that only part.
Cell membrane. Like, I get why it's C, but can we just go through why it's not D? Because I figured...
So when they said the adherence of neutrophils was facilitated by an adhesion receptor on the neutrophil membrane, a neutrophil is a cell, and its membrane is a cell membrane.
Okay.
So if it has a defective cell membrane, it wouldn't have a working adhesion receptor on that membrane, right?
Oh. Okay. Yeah. That makes a lot of sense.
Yeah. So basically, like, it's working fine. Like, if it can still migrate through the endothelium, right?
Oh, yeah, so it's working fine, so we don't need anything defective.
Mm-hmm. So, yeah, it'll just be C. It can still bind to the endothelium. and it's probably like a missense mutation or something, like one of those mutations that just causes like either one amino acid to change into something that's similar, or something like that. But it's not really, it's not like a loss of function mutation or anything like that.
Yeah. So it's probably like.
Mm-hmm.
So this like passage made a lot of sense now that we went through it slowly.
Mm-hmm.
But I guess like practice is probably like one of the only things. Well, genetics, this one like threw me off a lot because like the whole XXXY. Like,
Yeah. They always do that. That's why I like doing like the shorthand stuff. So okay, let me see. So it seems like that question is talking about. All right, I'm just gonna like go through the passage, or whatever. So okay, the most common form of hemophilia. Yeah, what's up?
Are you saying we should go through this first then?
I mean, it's pretty short, and since they're asking about the factor 8 gene, and they're talking about it from like the first sentence, I'm just gonna go at it.
Okay.
So okay, it seems like the most common form of hemophilia is caused by a deficiency in the functional form of a soluble blood protein that is essential for blood clotting, which is factor 8. Okay. All right. It's in liver. Okay. Since it's asking about, let me see. I have a hypothesis states that all mutations leading to hemophilia occur within the factor 8 gene. Which of the following pairs of inheritance patterns would fail to support this hypothesis? Okay. All right. So let me see. All right. Nothing else in the first paragraph is talking about the gene, so I'm gonna like kind of skip that.
Okay.
Second paragraph talks about the gene. All right, it's 186,000 nucleotides long. By the way, the speed of light is 186,000 miles per second. Anyway. the factor 8 protein is only that many. Okay. All right. Introns, blah, blah, blah. Okay. All right. Genetic defects leading to hemophilia may take the form of either nonsense mutations or deletions. All right. So that kind of helps us. All right. It's on the X chromosome.
Wait, where are you looking?
Oh, that was like by the end of that second paragraph. But the third paragraph basically tells us the answer, right? It's a sex-linked recessive, like disease.
Yeah, this is like my freshman year bio, so it's like that, like freshman year bio, freshman year chem. A little rusty on for sure.
Oh yeah, so basically, if it's sex-linked recessive, it means that,
They're recessive, and then I know like the men have XY, so then like if it's recessive X link, like the man has to have it. If
yeah, because the woman would need to be homozygous for it.
Yeah, no, I get that.
Yeah.
So I think I get it.
Mm-hmm. So okay. So since the hypothesis is saying that all mutations about hemophilia. So that just means that everything that they're telling us about hemophilia we can use for the factor 8 gene. So let's just equate factor 8 gene to hemophilia, right?
Wait, so we're saying which would fail to support the hypothesis. So we want to fail to support.
I would say. Oh, sorry. Say this again. What are you saying?
Or we want to fail to support that it's an X recessive.
Yeah. So I think maybe you chose the thing that would support it, right?
Wait, so normal sons being born, wouldn't that fail? One.
Normal sons born to hemophiliac fathers. No, that would make sense because a hemophiliac father, right, would have an X, like an affected X chromosome, right?
Yeah. So then.
And then, yeah, sorry.
Oh, but then the mother
could just be a carrier.
Yeah, so the mother could be, or she could be like completely fine, right? So
yeah, that's true. The mother could be completely fine or heterozygous. So, that could be something that works, right?
Yeah, I think I only like took into account for that.
Mm-hmm. Yeah, because that would normal sons born to hemophiliac mothers. So
Would you have like X and then like HXH in order to be affected since it's recessive, right?
Mhm. Yeah,
So, and you would have to get this.
and then yeah, and then the father could be regular X Y, right?
Yeah, but then he'd still get one of hers, right?
Um, yeah. So that basically wait, which of the following pairs would fail to support this hypothesis?
Oh wait, but you're right. So like, what if the father is like just regular X? No, he'd still get one of. I don't know. How would that work?
Oh, you can put it like as a Punnett square, like vertically.
Yeah, so I know how to like do this, but then oh, because then never mind. So 50 of them would be girls and 50 of them would be guys. I get it. So then this would be XY. Oh, that was supposed to be an X. So then yeah, he'd have hemophilia in both cases, right?
Mm-hmm.
Okay, so this would fail, right?
Um. Yeah. Mm-hmm.
Hemophiliac sons born to normal fathers.
Let me see.
That would mean that the mother had to be.
Well, that's like the same example we're using for that previous one, where the father's normal. So that actually supports it.
Wait, 3 is the same as.
Yeah, because we have a normal father there, right?
Oh, you're saying here.
Yeah, the one above that. But yeah, same. Yeah, where you drew that Punnett square, that's a normal father, right?
Wait, because. But then what if like the mom's just a carrier for this one? Like I feel like this doesn't give us like enough info.
Oh yeah, it's just saying that it doesn't give you enough info, which means that it's not necessarily true, right? Like hemophiliac sons, born to normal fathers, right? That uh, can happen, and it doesn't have to happen.
Oh, but this one, because this one's not necessarily true either.
Oh yeah, because the daughter will receive a X chromosome from the father, and it has to be a regular X chromosome. So that can't be possible.
Wait, I thought it. Oh yeah, okay, yeah. No, that makes sense. So it would be 2 and 4, right?
I believe so.
Okay, I have to accept the annotation. Yeah.
Yep.
Okay, that makes sense. You're gonna get. Okay, see, I got right. Oh, that was easy. That was easy.
Mm-hmm.
I just want to like find one with one of the graphs I know gave me like a lot of trouble.
Mm-hmm.
Oh, this is like a weird question. Why does I get the difference like in base pairs GC has 3, but why does PCR need high GC content?
I think it's which primer is most suitable for PCR? Well, okay. First of all, GC content means that it can sustain higher temperatures before melting.
Oh, so it would just be more stable because the extra hydrogen bond and like PCR is under high temperatures.
Yeah, and GC can make 3 like hydrogen bonds
Yeah,
as opposed
instead
to
of
2. Yeah.
Okay, I just have no idea. I gotta go over like this non-disjunction, meiosis, all that.
Oh, non-disjunction just means that when 2 homologous chromosomes, they line up at the metaphase plate, right?
Yeah, I mean, I really have to go.
Yeah. And then they get pulled to opposite ends. So non-disjunction is just when, like, you know, each homologous chromosome gets pulled to opposite ends, but let's say both of them go to one end, that's non-disjunction.
Oh. Okay. So this is when, honestly, this I can't even like begin to process because I really don't know.
Oh yeah, just think of it like this. So like, it's prophase, metaphase, anaphase, telophase. So prophase is basically just when, we got like the nucleus disappears and the DNA turns into like chromosomes.
Mm-hmm.
Then it's metaphase where the chromosomes line up in the middle.
Yeah.
And then it's anaphase where the chromosomes are pulled to opposite ends.
Okay.
And then telophase is when, the cell, because they have stuff on opposite ends, actually turns into two cells.
But then there's a whole, like, two.
Oh, okay. So with meiosis, that happens twice.
Okay.
But since they're giving us just one type of anaphase, and one type of metaphase, and one type of prophase, and one type of telophase, right?
Yeah, no, honestly, I know you didn't need it for this question. But
Oh, yeah. But I see what you're saying. Like how, there's...
This is definitely just...
Yeah.
Yeah.
Yeah. It also helps to remember that any, two... Like anything with the Roman numeral 2, is the same as mitosis essentially.
Is the same as what?
Oh, it's the same as mitosis.
Oh, like the steps are the same?
Yeah.
Oh, yeah, that will be helpful.
Mm-hmm.
I remember hating that. Wait, let me see if there's... I know this one kind of gave me trouble, but I want to go...
Mm-hmm. We'll be right back. I'm just going to use the bathroom.
One. Okay.
Okay. What school do you go to, by the way?
Hofstra.
Oh, you're in New York?
Yeah, where are you?
New York.
Oh, really? Yeah, I'm on Long Island.
Oh. Yeah, well, yeah. I grew up in Long Island, actually.
Oh, really? Where?
New Hyde Park.
Oh, okay. That's actually right by Hofstra.
Yeah. Did you grow up in Long Island?
Yeah. I'm in Greenlawn. It's near Huntington.
Did you say Greenlawn?
Yeah, you know Greenlawn.
Is that in Suffolk?
Yeah.
Yeah, one of my best friends lived there. It's out there.
Yeah.
That's pretty funny. Yeah, I'm in the city now though. Okay.
Oh, nice.
Yeah. All right. Yeah. This you mentioned this about red blood cells.
Yeah, it wasn't this. Just like when I see graphs, some of them I have a hard time reading.
Yeah, the graphs and charts for BB are definitely the most annoying. They're just really messy. They're it's just full of crap. I always hated them.
Pick a specific one. I mean, I guess this one. I don't really know. Just some of them. I don't even know. I'm just seeing the T.
I see retrograde trafficking again. That can actually answer that question before. See how it says to the ER? I'll be right back.
Retrograde trafficking.
Yeah, it says to the endoplasmic reticulum.
Yeah, you actually totally can.
Yeah, sometimes stuff from later passages can help.
But honestly, this is by the end of the test. I would not have
Oh yeah, that's why I have like a very like, I'll tell you this like way late, like right before your test, but like I like to like the way that I like time it is that I want to I like flag very liberally, like if anything is that I'm unsure of. So by the time I see the last question, I should have like 20 minutes left, and then I can, yeah. But that means I go through every other question like not like completely, right? But it's just so I because if you treat everything like you can't treat all the passages equally, just like you can't treat all the questions equally because they range in terms of difficulty. So,
Yeah, no, I get that, but
so if I do that normally, then there's a high chance that I can have like an especially hard passage early on.
yeah.
And so I just started like at some point I started to do that when I was taking this test, and it just like changed a lot.
So then, would you like see how this is like 52 to 56? You wouldn't do all questions during, like right after reading it? Because I feel like if I skipped around, I'd get it confused with like other passages or just like have to reread it.
Oh yeah, I would like yeah, I'll tell you more about this like towards the end, but basically like I would start by doing the discrete problems first.
Oh. Yeah, I mean, because I'm supposed to take the Kaplan diagnostic,
Yeah.
I think, next week.
Mm-hmm.
So hopefully I like do better, but I don't know.
Yeah. I basically would like write down which questions would be the discrete questions. And so when I start the test, I would just do the discrete ones first.
But how do you like? Do you just like go through looking at them?
No, I would go to like the navigation page or whatever, and then just click on them. So I would do those, and then doing those first like makes me go to the first. Then I go to the first passage already having the confidence that I have answered like whatever like 12 questions or however many discrete questions there are.
Yeah.
And yeah, and if anything is taking me too long, I just like flag it, or if I'm not 100% certain about a question, I would flag it, and I would just go through like it as fast as I could, so I get to like actually like see like all the passages at least.
Yeah, because I know Kaplan tells us to like be careful with like what you flag, but I know I flagged like so many questions when I did the practice test.
Yeah, when I saw that, I was like, okay, good, because that's how I do it. So, all right, I see a lot of crap. Like, let me see the question so I know what to look at.
Oh, sorry.
It's okay.
This
Most likely result of various alanine to proline mutations. Okay, so right away, I know it's going from something that has very little steric hindrance because alanine is tiny, to proline, which is
a breaker.
Yeah. So, okay. Now let's see why. So, all right.
So,
yep.
For like alanine, like what are we looking for?
Oh, we're looking for, I guess, the reason that it changes, right?
Yeah, but like, are we just gonna read this whole thing then, or are you gonna like? Should I go back and look for like?
Oh, I'm just looking for like the word alanine or proline.
Okay.
I don't really see it. Let me see what else I can look for.
See them either.
Helical, okay. And then
oh wait, alpha helix. Proline's an alpha helix breaker.
Yeah.
So wouldn't that
Loss of secondary structure.
Okay, now I get that. If you're breaking your alpha helix, you're losing your secondary structure.
Yeah.
I guess I just need a question on like the graph. Several strains. What?
Several strains of STX producing E. coli are responsible for disease. Despite the availability of antibiotics that lyse, aka, cut in half, E. coli
Yeah.
cells, the reason this is not... Oh, okay. Probably because it's going to kill, like, the good ones, I think. Let me look at the STX thing. Like, what exactly is that? Okay. Okay, it's basically a toxin, okay. Wait. Alright. Okay. So it seems like it moves around.
What are the choices? GPP is also target. So you're saying you think it would be B?
So it's not going to be anything involving viruses.
Yeah, well, this one like kind of just seems like a weird statement, so I feel like I'm comfortable getting rid of that now.
Yeah. Because E. coli are bacteria.
Yeah, so then STX confers antibody resistance once inside the whole cell. I mean,
So, okay, they're basically saying that antibiotics won't work.
that would make
say how it moves around, right? Like, basically, in that first paragraph, or maybe second, it talks about... Oh, I guess there's only two anyway. But it's... So, okay, they're basically saying STX producing bacteria are bad, because STX is bad. It's a toxin. It's made out of these two subunits, right? And basically it moves from the cell surface to the ER. And then, let's see. Okay, wait. Okay, so subunit B mediates trafficking from the cell surface to the ER. Then A turns into a catalytic domain that works by depurinating a specific position on the ribosome. So okay, subunit A works to stop protein synthesis. Okay. Then a different part of subunit A stays with subunit B, okay? Then it was thought that GPP130, which is a host membrane protein, which means that it's on the cell membrane, okay? Cycles between the Golgi and endosomes, okay? It might assist STX in evading degradation and reaching the endoplasmic reticulum as it binds the toxin. So, okay. GP, whatever, binds to STX. Okay. Now, then they do an experiment to see if GPP helps STX move from the endosome to the Golgi, alright? They made STXB, which has subunit B that, that fluoresces, that gives off light. So we can measure, like, based on how much light it gives off, how much subunit B is around, right?
Mm-hmm.
Okay. Then they measured the percentage of STXB in the Golgi, alright, in human cells, and then repeated it with a fluorescently tagged cholera toxin, which is CTXB, and that's in figure 1, which follows the same route as STX, but does not require GP. Okay, got it. So okay, first of all, let's see. Okay, so left is STX, right is CTX. CTX needs GPP. STX I'm sorry. CTX does not require GPP, but STX does. And then so the control. Oh, and then anything with so if the p-value is greater than 0.05, it's basically not statistically significant. So if control and presence of manganese, right? Manganese degrades GPP.
I feel like I have no idea.
So okay, here, let me do this. I'm gonna take a screenshot and then I'm gonna like draw some stuff.
Okay, let me know when to stop sharing.
Okay. All right. You can stop sharing. I'll share. All right. So here. Okay. So all right. So basically. Okay, so manganese. Oh, should have used black. Manganese basically. Oh, I don't know why that just disappeared. Okay, let me open whiteboard up again. Okay. So, all right, manganese, I'll say, is inversely proportional with the amount of GPP. Because over here it says exposure to manganese degrades GPP.
Okay, exposure to. And GPP is the host membrane for STX.
Mm-hmm.
Okay. So you're saying manganese has an inverse.
Yeah, and STX, right? It needs GP. So they're basically trying to figure out whether GPP is needed for STX, and we know that CTX, cholera, follows the same route as STX, but does not require GPP.
Wait, if it's the host, this is probably a really dumb question, but if it's the host, wouldn't it need it?
Sorry, what are you saying?
So isn't GPP the host for STX?
It says host membrane. It's just a protein.
Oh, so it's not like the host for. Oh.
Yeah, host membrane protein. Yep.
So what is a host membrane protein? Like what's the host part?
Oh, so I guess the bacteria, right, guts into like a host like cell, like maybe a human cell.
Okay.
And on that host like cell membrane, there's a protein called GPP.
Okay, so it's not like the vectors, nothing like that, like a host vector.
So let's say this is a human cell, and this is like the lipid membrane. So this over here is GPP. It's just a host membrane protein.
Oh, okay. All right. I'm thinking something different.
Mm-hmm. Yeah. So basically, we know that manganese degrades GPP, right?
Mm-hmm.
Now, we have two experiments here, right? And there's like the triangle means it's a control. The whatever circle means that manganese is added, so GPP is going to be all effed up. Okay. Now, we know that the cholera, the CTX, follows the same route as STX.
Oh, but it doesn't need.
But yeah, it doesn't need GPP, right? So that's why when you expose the CTX to manganese, it's which degrades GPP. It's basically the same.
Wait, so what's the like what's the thing on the left? Oh, okay, wait, yeah, no, I totally get it then.
Mm-hmm.
Because then it doesn't if it doesn't need it, then it's gonna have the same activity.
Yeah.
But then so basically we just found out that STX is needed, like needs GPP.
Correct. Because for
Okay,
STX
yeah, I got
with GPP, it's in the Golgi, right?
Yeah, okay, wait, that makes a lot of sense.
Mm-hmm. And then without it, it's barely in there, right?
Yeah.
So I wrote like the amount of manganese ion, right, is inversely proportional to the amount of GPP.
Yeah. Okay, no, I get your notes now.
Yeah.
I think like
The amount of STX, I should say the amount of STX in the Golgi is proportional to the amount of GPP.
yeah, I get that now.
And then the amount of CTX is just, there's no relationship between that and the amount of GPP.
Okay.
Yeah.
I don't know. It's just these sometimes like, when I see a graph, I think even if it's like not hard, because this wasn't hard when we like kind of just broke it down, but when I see it, I'm just like, holy shit.
Mm-hmm.
So I guess, do you think like the best way to do these to get better at these? Because I feel like these are the ones that I'm most like, holy shit for.
Mm-hmm.
Is just like so much practice, but then it's weird because I feel like the biochem books, like obviously they don't really have that much practice with these, like you know.
Oh, I
they're
just wanted to say like one last thing, which is this drawing,
Oh.
because over here it says STX, right? It basically it has the B subunit, which makes it move from the cell surface to the ER. So let's say this is like whatever cell, and this is like the cell membrane thing that I drew around it.
Yeah.
And so this is STX right there. Right?
Yeah.
But it goes back into the ER. So basically, like, if we have, like, whatever, an well, that's for antibody, but, like, whatever, an antibiotic, right?
Mm.
And it's going to be looking for this STX that's on the membrane. It's not going to be able to find it because it's the STX is going to go into the ER. It's going to be, it's going to go back inside of the cell. So the antibiotic is not going to be able to tell if a cell has it or not.
So that's the answer to like that's like the question.
Yeah, so you can go back to sharing.
I was like, where are we going with this?
Yeah.
Okay. Okay, so then you're saying the antibiotics. Oh, and you're just saying it won't. It's in the ER. Okay.
Let's see. The bacterial lysate will disrupt host translation.
Actually, no.
Wait,
I
yeah.
thought it's.
All right. So, okay, this isn't necessarily okay. Let me see. Several strains of STX producing E. coli are responsible for disease. Despite the availability of antibiotics that lyse E. coli cells. Oh, I think okay. I get it. So basically, what I drew, saying that the antibiotic won't find it, is irrelevant because they're saying that there's antibiotics that will completely cause a cell to like explode. So if that STX goes inside of the cell, right, in the ER and whatever, and we break that cell open, that STX is just gonna be out of the cell, and it can do more damage.
Right.
It's like yeah. It's kind of like the lysolytic cycle of a virus. I know this isn't a virus, but like, you know what I mean? Lysogenic cycle, lysolytic cycle.
Kind of, yeah, but
Mm-hmm.
so I understand what you're saying. Like, you're allowing it to come out, so it's gonna do anything. But how is that like? How is that this?
Have you ever seen the movie Alien? It's kind of an old movie.
I don't think so.
So it's like this type of alien that has acid for blood.
Oh gosh.
So it's like if you like try to kill it, it's just going to release that blood that's going to kill you. So they're saying that we have antibiotics, right? That lice, that basically cause, E. coli to like break apart and be cut open and stuff. The lysate is basically the stuff that when you lyse something, it's what's left.
Wait.
So when they say the bacterial lysate, they mean like what residue is left after you lyse it, after like you cut it open.
So if you cut it open,
Mm-hmm.
and like, wouldn't it just be straight though?
So
Or like this?
yeah, if you cut it open, because look, the thing with STX is it is able to hide in the sense that it can go from the cell membrane to inside the cell.
Yeah.
But if you then destroy the cell and it's inside the cell, it's just going to be out and about and free.
Wait, so you're saying the bacterial lysate? Oh, so you're saying I don't get the lysate thing.
So the lysate is just like another word of what is like, how should I put it? It's like the so okay, let me say that I give you a cell of E. coli, right?
Okay.
All right, now let me say that I caused the E. coli cell to lyse, like to be, split, and all of its like guts come out. And then I give you that. That would be the E. coli lysate.
Oh. So if you kill the cell,
Yeah.
then the STX is going to be released and kill everything.
Yeah. Because, the STX is, because it's basically, it's like in an endosome, right? Which is like a vesicle inside of a cell.
Yeah, that makes a lot of sense to me. I just, like, it makes sense that I'm going over it with you, but
Yeah.
it's going to take me a lot of practice, I feel like.
Well, yeah, but it'll start to like get like easier as time passes.
Yeah, it's just weird because I feel like a lot of when I was shocked when I took my first practice test that like I feel like there's so much information here, but like a lot of it I feel like isn't even tested. A lot of it you can get from the passage or
Yeah, definitely. So far we've used mostly information that is in the passage.
Yeah, like I feel like we use only information.
Yeah. So that's why, like, I don't understand what purpose there is in knowing, like, just doing pure content review, because in my opinion you've already learned much of what's going to be tested because you took like the classes already.
Yeah, like I feel like for a lot of this stuff I don't know, the bio when I did my first chapter bio I was kind of like in for a shock, but the gen chem I definitely forget, and the physics I feel like Hasha at least like I feel like I just learned physics in a very different way, and like I know a couple friends who took the MCAT but like ahead of me, and I don't know, just the way we learned physics was like weird and not at all how they test us, so I don't know.
Yeah, I mean, like, there's like a reason why like physics majors score so high on like on like the LSAT, the MCAT, the GMAT, the GRE, etc. It's just because it forces you to think in a way that is focused less on what you know and more on what you can figure out.
Yeah.
And yeah, and so, you know, and you're kind of like seeing that now, like how we kind of just like look at the question, we can, yeah, like I suppose like figure things out based on.
Yeah, it's like weird. I was really shocked because like, but then I'm just like, why am I doing all this content review if like maybe
Mhm.
there's really not that many like straight content review questions in the exam.
Yeah, I would say only do content review as you, like, in a targeted way, like on a need to know basis. Like if you know, if you come across like, I've only seen that mostly for like maybe like biochem, like metabolic pathway questions or something like that.
And how specific though, like, will those be?
Well, I mean, like, there's different ways of like approaching it, I suppose. Like, for instance, you can memorize every single pathway, which is not how I would do it, but
Yeah, now that you're
I would
terrible.
say I would say to focus on like the irreversible path, like the irreversible steps, but ask yourself why. Like, why is this step irreversible? Because
Oh, and that's like we needed to know that for my biochem class.
okay, that's good. So you're on the right track then. But
But that's also a lot of Oreo, I feel like, just like if you know the structures.
exactly, yeah. And since you TA that, like, you know, like how, like, you can predict how because like biology is like applied chemistry, which is like applied physics, which is applied math. So if you have an understanding of, like, if you can just look at a structure, right, and see like a benzene ring or something like that, you can, yeah, you can you can deduce a lot from it. Like, you can kind of, like, you don't need to memorize the ones that are non-polar because you can just look at it.
Yeah, that definitely helps a lot. Just like the physics and the mental math is like really
Oh,
really
so okay, the math stuff is okay. That's important for us to do, because, like, even like, though I say don't memorize like things, there are like math things that come in handy to memorize.
yeah.
Like doing things like
Anything without a calculator, so it's just like weird.
oh yeah. Yeah. So is there like a math like question that you have like readily available? I can kind of like show you.
I can look at like the physics. I can pull up like my physics and math. Like it took me, and then it's just weird because then I'll like look at their explanation of it and I'm like, oh, like that makes sense. But obviously makes sense like after I'm like staring at it for like 30 minutes.
Yeah, and sometimes like the explanations aren't even like, so I mean, there's like a couple of things that I want you to memorize in terms of math. Like, memorize like certain logarithms. Like, you'll just be able to see lots of patterns and stuff.
Okay. There's got to be some math on here. Let me see.
Oh yeah, there should be like a bunch in that practice test that you did, but probably in like the CP section.
Which ones I did? Wait, in the what section?
Like the CP section of your sample test.
Yeah, this is one of the CP. I just did this one yesterday. Or 2 days ago maybe.
Yeah.
Quite wrong.
What, the Kaplan thing or the
Yeah,
CP
these are like the
section of your sample test.
Kaplan end of section quiz
Okay.
for the
Mhm.
kinematic dynamics.
Okay. But in the sample test that you took for the CP section.
Oh, that's a good question. I like always forget how to get there. So hold on.
Like the prep hub.
Yeah, I feel like there actually like wasn't that much math for that I wasn't like
Mm-hmm.
too like confused about because it would be like a relationship where I'm like, okay, well I know like it's gonna be lower or something.
Yeah.
Um, but oh wait, let me stop sharing and share this.
Mm-hmm.
Wait, so do you just like tutor now, or are you in med school? Like, what is
I'll tell you about that in a sec, okay? I just want to.
how do I get to? Oh,
Oh yeah, you could do like whatever review all, or I guess you could click that and then hit review all.
okay. So, and then should I just
Yeah, you can like go yeah. So but yeah, if you're doing it like that, you don't get to see the question. So I don't know if you'll.
Oh, I don't think there was math in like a lot of the passage ones though.
Okay, I saw something with like some numbers. So all right.
Oh, you did? I can go back. Or like,
Okay, I mean like we could even do this one.
is this even?
This isn't like really like intense math, but
Wait, I can.
Mhm.
Let's see. I didn't think there was math on.
If you do like command minus, I think it'll let you like zoom out a bit.
Oh,
Yeah.
a lot easier. The pH I know how to do.
You want to see? All right, I was gonna show you something, but let me see. Oh yeah,
Up here.
for this one, I thought there might be some because I see like I see radius and stuff like that.
Okay, so not on this question, but probably. Oh, here?
Yeah, that seems to be a math one.
Okay. So, do you want to do this one?
Okay, let's see. Closest distance the electrodes use. Okay, NCV test. Okay. Let's see. Two electrodes are placed on the patient's body. Electrical shock placed on one, and the nerve response measured on the other. So the pulse travels at 100 meters per second. Okay. Closest distance. So basically, if speed is a change in distance over time, you're just gonna need to find the time. If you multiply it by the time will cancel out, and you'll get your answer in meters.
What is the closest distance? So then.
It tells you there that it takes wait.
Oh, the speed you're using?
Mm-hmm.
So we're using the speed, and then what time are you using now?
So it seems like on that thing, a typical nerve impulse. Let's see.
Up a little, or?
Oh, just figure 2.
Oh. Oh, one. Oh my god, I didn't even like see that.
Mm-hmm. Yeah.
So that's
yep.
A nerve impulse is 1 millisecond.
It seems like each of those like things, like each of those like little like dashes or ticks rather on the horizontal axis is like the distance between one and the other is 1 millisecond.
Wait, where are you looking? This is what you. Yeah, I see. Like, you're looking at this, right?
Yeah, and so I want you to start using. So you know, like on a calculator, instead of saying 1 times 10 to the negative 3, it just says 1 capital E neg 3.
Mm-hmm.
Okay, so like that is how I want you to like write it out from now on, because it makes things so much faster. So
Okay.
when I see 1 millisecond, I just think 1 e neg 3 seconds.
Okay.
Yeah. And 100, I see 1 e2.
Okay.
So then if I were to yeah, so basically I see that, so they're saying that an NCV test basically involves 2 electrodes placed on the patient's body with an electrical shock placed on one and a nerve response measured on the other. So I see that nerve response, aka the nerve impulse, aka the action potential, like basically that spike, right? That's just within 1 millisecond.
Yeah. I mean,
Mm-hmm.
so
So, yeah.
I see milliseconds, and maybe I'm just so this is telling you that like the distance from here to here is 1 millisecond, but then
Mm-hmm.
I'm like, well, this kind of looks like it's like less than. Am I like looking at that wrong?
Well, think about where it's starting down there.
oh,
Like, so that's where it first, gets
so it's like
that,
from
right?
here.
Yeah. And if you like bring those like straight up, it's exactly 1 kind of millisecond.
Yeah.
Yeah. So I would write 1 e neg 3 times 1 e2.
So 1 e neg 3.
Oh, I guess seconds.
Yeah. So seconds.
Mhm.
Divided by.
Well, all right. If we're using speed, which is meters per second, right?
Yeah. So wouldn't it be distance over time?
Yeah, speed is in distance over time, and we
Oh.
need distance. Mhm.
Formula. Yeah. Okay.
So we have to multiply it by time, right?
Yeah. So it would be speed times time. So that would be the 1 e. Okay, I see what you're saying now. Yeah.
Yeah. So I would write the 100 as 1 e2,
Yeah. I did that.
and then
So then
multiply by 1 e neg 3.
and then you get 1 e negative 1.
Yeah, because with exponents, if you multiply them, you add. If you divide them, you subtract. If you take one exponent to the next exponent power, you multiply. So yeah, your answer would be 1 e neg 1, which is 0.1, right?
Yeah. Okay. That wasn't bad.
Mm-hmm.
That really wasn't bad. I don't know. Some of the math just like gets me. I know in the practice book there's like decimals and fractions.
Oh yeah, use fractions as much as you can.
Instead of decimals?
Yeah.
Okay. Like I know in the practice book when it does the explanations, it just rounds the decimal either up or down every single time.
Yeah. But I think there's a lot of better ways of doing it. Like when I go over like some next step questions, which you may have seen in the PowerPoint, like their explanations are like they classified as like a brutal difficulty problem, and they use such an excess amount of math when it's so much easier doing it, another way.
Yeah.
Like
And then-
give me like a number as a decimal.
Just like a random number.
Yeah.
2.6.
So I would write that as 2 2/3, which is basically 8/3.
But then if it was like 2.7, like that's not as nice. What do you do?
So 2.7, I guess 2 7/10, which is 27/10.
Oh. Yeah. I don't know. It's like I guess all the equations is
Mhm.
like I don't know if I should.
Yeah.
I hear people have done it.
Show me some more, because like with equations you can figure it out using units, and if you don't know the units you can figure it out using the equation. But I can see. I'll like show you in terms of like examples.
Oh wait, I think I should zoom out again. Probably. No. I don't think.
Yeah. There was one that gave you the hydronium concentration and asked you to use find the pH.
Oh, pH I can do.
But that one had a different number as its mantissa, because it tells you 1 times 10 to the negative 5, and it's easy that way. But what if it was 4 times 10 to the negative 5?
That would not.
So let me tell you how to do this. So basically you take that exponent and you subtract it by the log of that first number.
Wait.
Yeah.
What?
So and there's certain logs that I'll send you that I want you to memorize. Basically like log of 2, log of 3, log of 5, and log of 7. If you know those logs, you can figure out the logs of anything.
Okay.
Yeah. So like this one would be basically 5 minus like 1.
So like the.
Oh, sorry. 5 minus log of 1, and log of 1 is 0, so that's 5. But if it was like 4, right? Like I'll show you how to do that, because then you start to use logs.
Okay. Yeah, I didn't even know that they could do that.
Mm-hmm. Yeah.
Oh, should I share my screen?
Oh, I was gonna say scroll some more, but yeah.
Oh, I thought you were showing me.
Oh, yeah.
Okay. Let's just tell me when to stop.
Okay. Well, did you understand that one?
This one?
Yeah.
What is this concentration of Cl minus ions? Okay, I actually do not.
All right.
I kind like I get this. I get it's like Ca, Cl, Ca is plus 2, and then Cl is minus 1, so there has to be twice as many.
Okay. Yeah, so
But-
it'll just be 2 times that, right?
So it'd be 2.
Mhm.
Then this is when I get confused. So
Yeah.
how could it be 2 times that? How could this be stronger than this?
Well, okay, so let's say you have CaCl2, right? It turns into Ca2 plus 2 Cl minus, right?
Mm-hmm. But then I don't know. I don't get how this can be bigger than this. Like I knew it was going to be something with a 2,
Mm-hmm.
but I thought it could this wasn't able to be bigger than that.
Oh, yeah, just because because calcium chloride, think of it as like a solid or something, right?
Mm-hmm.
And when it dissolves, or what about H2O, right?
Yeah.
Like that's going to turn into like H plus, like whatever, H plus
Oh,
O.
yeah. I know what it looks like when it dissolves, but
Mm-hmm.
I don't know how to ask the question.
You're saying how could it be like a greater concentration of Cl minus than calcium chloride?
Yeah, then the total solution. I guess I thought that Cl minus and Ca plus. I mean, in this case, I know my answer wouldn't make sense. That's kind of why I was a little confused on this question. But
Just think of for calcium chloride to exist, you need twice as many chloride ions as you do calcium.
so then if
It could
they
be like
ask,
a limiting reagent. Think of like limiting reagent problems, kind
Yeah.
of like that.
no, I definitely get that it's 2 plus, but I guess I thought that the Ca and the Cl would have to add up to this.
Oh, I see what you're saying.
Yeah, that's
I would just write CaCl2 arrow Ca2 plus and 2 Cl minus.
Yeah, I get the equation, but I thought the concentrations would have to add up to this, but I guess not.
Yeah. they're not going to be because you're making a compound that just in order for one calcium chloride to exist, you're going to need twice as many chloride as you do calcium.
Yeah. So then the concentration of Ca plus would just be 0.1.
Yeah.
Ca2 plus.
Yeah, that'll stay the same. Yeah.
Okay. Yeah. No, I guess I get that. This I got. I just need to. So this is like a thing. I guess I need just the charges of the amino acids, right?
Oh yeah, but again, the only ones that'll have charges are the acidic and basic ones.
Yeah.
So, and the basic ones will be positively charged. The acidic ones will be negatively charged.
Wait. So which? Arginine, right?
That's the
Okay.
Because if you think about it, like if at a regular pH, right, or they said pH of 8,
Regular, I feel like.
yeah, if something's more acidic, it's going to be in a basic environment, so it'll be deprotonated. And if something's basic, it'll be in an acidic environment, so it'll be protonated.
Yeah, I get that. I get that, but for biochem, I feel like we had the charts, so it was a little bit easier. That doesn't look like. This looks like math, but I seem to have gotten it.
Oh, okay.
I actually kind of remember.
So something that's a little weird here is converting cubic centimeters to cubic meters.
Oh, that's why I struggled.
Yeah. So okay, that's a little weird. So basically, think of it like this. So okay, well, 1 meter is equal to 100 centimeters, right?
Yes.
So then 1 square meter is going to be equal to 100 square centimeters.
They just add 2 zeros.
Yes.
Okay.
And then a cubic meter will be equal to 100 cubic centimeters.
Wait, so you're doing 1 meter, 100. Let me just write this down so I can visualize it. 1 meter square is 100.
Yeah. Or
centimeters.
1E2. But yeah.
Oh, yeah, wait, I should practice that. So, wait, I don't think I'm writing this right. So 1 meter is 1E2 centimeters. 1 meter squared is 1E4.
Yeah.
1 meter cubed is 1E6.
Yeah.
Okay. Yeah, so I think that's like the only thing I didn't get on this, but I understand like density is like pretty like self-explanatory.
Yeah, and if you think about it, a cubic centimeter is the same thing as a cc. That's why they call it cc, which is the same thing as a milliliter.
I didn't know that. Yeah, I didn't know that at all. See, like, it was weird. I was kind of shocked that there wasn't, like, too much math on the practice exam.
Yeah.
I don't know how similar this is to, like, the real one, though.
Yeah, there's a company called Next Step, also known as Blueprint, that they have really, like, calculation-heavy science sections that I think is really a good practice. Oh, that seems like one. Because on the actual test, like, there's always going to be more physics and more calculations. So.
What you think?
Yeah.
Then like these practice tests. Oh, that scares me.
Yeah. So, I'll definitely make sure that you see your fair share of math stuff. Okay, let's see. Density of the wire at 473 Kelvin. I look at the chart. I get the length. I get the resistance. I get the current. I get the voltage. And then they tell me.
Yeah, wait, what?
Yep.
So density is mass over volume. But then what mass do I use?
Wait. Sorry. What are you saying?
Wait,
Yeah.
I know the density is mass over volume. Am I just supposed to use this mass and this volume and nothing from this chart?
From that chart, you're gonna use a different length. It seems. Let's see. At room temperature, okay, which is 293. The length is that, okay. Diameter is that, okay. Mass is that, okay.
Wait, what equation are you using then?
Oh no, I'm just looking at the top, and it seems like what they're doing is. Wait, an iron wire with known electrical and thermal properties, okay. Okay, iron wire. Change in length, okay. Barely a change in length. Energy radiated. Okay, the volume of the wire is 5e negative 7.
Like, the only thing I would have done here is I wouldn't have looked at this chart. That's what I'm saying. I would just do like M over V and then use this and this. Is that wrong?
Let's see. That would give you uh 0.8, or is it 8 that? Yeah, it's 8,000. Okay, yeah, that would work.
Yeah, so then what equation were you going to use using the chart though?
It seems like nothing. I just looked at the chart first because they gave me 473K Kelvin.
So it would be 0.8E4 I got, and yeah, take that. Oh, okay.
Yeah, so you should know like 4/5 is 0.8, and then neg3 minus neg7 is just neg3 plus 7, which is just 7 minus 3, which is 4.
Yeah.
But yeah, you would just use the regular density for that one. I was just looking at, because this is talking about like whatever, this thing called like whatever. As something gets hotter, the length gets a little longer. So, but it's essentially the same here. So it's whatever. So you use the right one. Oh wait, that one I think is a little important because if they're talking about an inverse relationship.
Which is
JK. Never mind.
No?
I'll talk about that some other time.
Okay. This is calculations. Oh, but this is QMCAT, right?
Yeah. It's a difference of 200 in terms of temperature. So you would just, I suppose, multiply it. Well, wait. Difference in 200 in terms of temperature, the amount of heat, energy. Yeah, I guess so. You have the weight, which is 4 grams, essentially.
What are the only equations I remember from physics?
Yeah.
Well, that and the kinetic energy.
Mm-hmm.
Basic one. I don't know if this is math or not. 5. No, this isn't math, right? No for this. I don't need math for that. They got a 0 again. Oh, that's the IR spectrum, yeah. I think there's not going to be any math in this passage. This equation. So look, so you're thinking like you saw many math questions run this, and there'll be like more on the MCAT, you're saying?
Yeah, I'd say so. Because, you know, if you think about it, everyone who takes the MCAT has taken these, you know, tests. So they're obviously going to be, you know, different. And usually when I ask them how their tests went, they usually say, you know, there was more math or more physics on it than the practice material.
Yeah. That's like my biggest fear.
Mm-hmm.
What is the frequency? I just learned this equation. So
HF or HC over.
No, this I think this is F. Oh wait, frequency emitted by the gamma photons. Wouldn't this be E equals HF?
Yeah. But you're gonna have to do another conversion here using coulombs. Well, unless they give you the wait. Oh yeah. Because they're just giving you the energy of the photon. I mean, so okay, you are going to use E is equal to HF. Because they give you E, but they give it to you in kilo electron volts. So you're going to have to make a conversion there.
Wait, where are you looking?
Oh, it says emitting 140 kilo, like keV.
Where?
Move your cursor to the right. Yeah.
Oh. Okay.
Yep.
Yeah, I think I used this over this. But you don't need this then, do you?
If they scroll up, see if they give you anything relating electron volts to anything else. Let's see. Oh, maybe the chart. I mean, the graph. No, that doesn't tell you.
Okay, so I guess they gave me that to confuse me. It worked. Okay, so 140 keV. And then what do you have to convert?
I mean, like, okay. I mean, that'll be. Let me think. In electron volts. Like, because basically they're giving you energy in electron volts, and they're giving you Planck's constant with joules. So you have to convert one into the other. Maybe the 30 watts can help, because that would be 30 joules per second.
30 watts.
But electron volts, I'm surprised it doesn't really tell you.
What's KeV to EV then?
Oh, I mean, that's just the kilo part. So it'll be 140 E3 electron volts.
Now, should I write it like 140 E3 or like 1.4 E5? Like, how would you?
Oh, it's faster to just do 140 E3, especially because things might divide into that.
So would I do 140 E3 over 6.6? So like, how would you would want me to do a fraction here, not just like round that to 7?
You would have to convert the electron volts into joules if you want to do anything with
Oh.
Planck's constant.
What?
Yeah. But they're telling you the elementary charge, and I think,
That's annoying.
yeah. Let me try scrolling up again, because usually they wouldn't ask you this without telling you. Let me see.
That's really annoying.
Mm-hmm. Alright, I'm pretty sure an electron volt is equal to 1.6 times 10 to the negative 19 elementary charge E is equal to this.
What they want us to do.
I mean, okay, F is equal to that, where H is Planck's constant.
But they don't want us to do any conversion. That's what's like confusing. Or they didn't write about it. How do you get B even with?
Mm-hmm. Yeah. Yeah, they suck at that. I'm pretty sure it's just like, I think it's like one. All right, I'm pretty sure it's like 1.6 times 10 to the negative 19. Either it's something like that with electron volts. Let me check. Oh, okay, got it. Yeah, it's it's that same value for the elementary charge. That value in joules is equal to 1 electron volt.
What?
Yeah.
Wait, I have no idea what you're saying.
Mhm.
So we have 140 E3 EVs, electron volts, and we have to convert this to joules.
Yeah.
And we do that by doing what?
So 1 electron. All right. So 1.6 e negative 19 joules is equal to 1 electron volt. I'm pretty sure because I've seen them make like a mistake before. I'm pretty sure that they meant to say like this thing is like that many joules or something instead of coulombs.
So, are we doing 1.6 E negative 19 times 140? E3?
So you're going to do so okay, 140 E3 electron volts, right? Times 1 joule, or no, sorry. Times 1.6 E negative 19 joules over 1 electron volt.
Yeah, you lost me.
Yeah, here. I'll do a...
Yeah, you're gonna share.
Yeah. How long are we doing this session for?
I really, like, whatever you want. Like, we can wrap it up after this if you want.
Okay. So yeah, basically 1 electron volt is equal to 1.6 E negative 19 joules.
So, okay.
So yeah.
140. Got it.
Yeah.
Okay. But you're saying on the MCAT they would give us this conversion, and I don't need to memorize this?
Yeah, unless they mess up. Because I've seen a question, I've seen like them mess up like this before. Like where they, like there's one question in the section bank that asks you for the number of molecules, which means that you would have to take the number of moles and multiply it by Avogadro's number, but all the answers are in moles. But yeah, so anyway, like So you would take 140 E3 electron volts and multiply it by 1.6 E negative 19 joules over an electron volt. But I don't like the 1.6, so I'm just going to turn that into 1 2/3, which is the same thing as 5 over 3.
1.6 equals 1. Mm-hmm. So then you're doing 5 over 3 times 140. See, even doing that in my head, like I can't. So how do you do that in your head though? 140 times 5.
So I was going to do like 5 over 3 times 140. 3 goes into 14 like 4 times with 2 left, and then 3 goes into 20, essentially like 7 times.
Wait, what?
Because 20, it goes into 21 7 times, but you know, and then I'm just gonna multiply that by 5.
That 3 and 4 thing, 14 thing.
Oh yeah. So I kind of like, so okay, I know this looks weird. But I get that.
Wait, I get. I don't. I get how you get to. I don't know.
So yeah, I basically just did. I changed the 1.6 into 5 over 3.
Yes. So I get how you get 20 and 2/3.
You mean the 20 over 3 down here?
20.
Because of the 6.6 is 20
Oh wait,
over 3?
no. I actually have no idea what you did then. Never mind.
Mm-hmm.
No.
So 6.6 is 6 2/3, which is 20 over 3.
Wait. I don't. Okay. I know, like, up here you were doing like 14. 3 goes into 14, then you got 4 and 2/3, and then I thought you multiplied that by 5 and got 20 and 2/3. That is what I thought I followed.
All right. Let's try it this way. Okay. How about this? 140 E3 electron volts, 2 E neg 19 joules over 1 electron volt times 1 second over 7 E negative 34 joules. And then I can just do 7 goes into 140 20 times to get 4 E. Yeah?
Where are you getting like the 7 from, though?
Oh, sorry. So instead of doing it like with fractions here, I'm just making the 1.6 into a 2 and the 6.6 into a 7.
That means it's 7. Yeah.
Mhm.
I get that.
Yeah, 7 goes into the 140 20 times. 20 times 2 is 40. And then I get 40 E18, which is 4 E19.
Yeah, that I think I get a little bit more.
Oh, yeah. That's usually what they tell you to do. Yeah, I guess, like, first of all, I'll send you basically, like, the math things to know. Like,
Okay.
this stuff.
Okay.
Yeah, and yeah, there's like a couple of things that I'll send to you that'll make like it easier to do it like this way, because this way I got like a much more closer answer, because the answer is like 3.38, right?
Yeah, I did notice like sometimes when I round, it's like the answer's not like extremely obvious sometimes.
Yeah, so yeah, that's why I use like the fraction thing here.
Yeah, but I think I could do it like way quicker if I round, but
Yeah. I'll show you like slowly, because I did it like pretty fast here, and we're dealing already with the confusion of the electron volt thing, which is AAMC's fault.
So I still don't like. Okay, we're converting electron volts to coulombs. All right. I get it the decimal way, the fraction way. I'm just a little confused.
Oh yeah, so we actually didn't use any of the Coulomb stuff because we basically just used electron volts, because they basically gave us energy in terms of electron volts, and they gave us Planck's constant in terms of joules times seconds. So we need to know what the energy is in joules, or what Planck's constant is in terms of electron volts.
I feel like I like really don't know when I need to convert things, so I feel like I need to work on that.
Oh yeah, but they literally made a what they're telling us here isn't like helpful, because they really aren't telling us like anything about electron volts. I had to like look that up over here.
All right, so then I won't be too confused about that.
Mm-hmm.
Yeah, so I guess the math, like that's all really the content for the things. I mean, I just, my biggest thing is like I really have no idea how to be doing this, but I think like that's it, question-wise, that I have today. I mean, I don't know. I just like, I feel like every time I'm studying, I'm not being like really that productive. So that's why I wanted to a tutor outside of.
Yeah, I would say like, all right, go through like the ones that you from that sample test that you don't understand still, and like send. Oh yeah, I'll make like a OneNote notebook for you, and I'll send you a link to that. But yeah, you can send me make screenshots of the questions you don't understand from that sample test and send them to me like in an email.
Okay.
And yeah, and then I'll try to like explain them in like a shared OneNote notebook. And if they still don't make sense, we could go over them next time.
Okay, yeah. I mean,
And then we can also go over like I guess like new stuff, but I would recommend doing like the section banks. Yeah.
when you said the section banks, are you talking about this? Like, I showed you all my AMC stuff that I didn't really like know I had.
Yeah. Do you want to share your screen? I'll be right back.
Yeah.
Oh yeah, I just like realized as I was in the bathroom that 1 volt is equal to 1 joule per coulomb. So, anyway, what were you gonna tell me?
Okay. I actually don't know how to find my resources unless I go through Kaplan's, so it's a sec.
Okay, I'm gonna fill my water.
Weird. The AMC website kind of confuses me. Find your baseline, then go to the AMC official test prep. Oh. Okay, I got it. Oh, sorry, I thought I was showing this one. Here it is. So, I don't know. Are these like what you're telling me to do?
Oh yeah, you can scroll down. You'll see section bank.
Or do I not
Yeah,
have that?
see it at the
Oh.
bottom. Mm-hmm.
So do these. But then
Uh huh.
you're saying don't content review first?
No, because when you do this, you'll see like what type of content you do have to review. Like, okay, so basically, first thing, like go through like the questions you got wrong on the sample test, and the ones that you got wrong, like see if it's like a content related issue or not. If it is a content, yeah.
Through sample test like the first time, I did that. And a lot of it was like content issue.
Okay.
Or like not understanding the passages.
Okay.
But I did write down like what the content was that I didn't get. If I
Yeah. So yeah, if you're doing content review, do the content review on the content that you didn't get on that sample test.
Okay. Like so should I not be reading these? Because I know I'm doing it wrong, even if I am supposed to content review. Because I'm doing a chapter day because I'm focusing on like the littler things rather than like the big picture.
Oh yeah, I'm saying like everything that I'm telling you to do is separate from what you have to do for Kaplan. Whatever you have to do for Kaplan.
Yeah, well, not like following along like exactly how they're telling me. Like if it's like pre-work, I'll do the pre-work and then kind of like see what I need to do.
Yeah.
But then they have all these like weird things that they want me to do after and before. And it's just like sometimes if I feel like it'll help me, I'll do it.
Mm-hmm. Yeah, whatever you have to do for Kaplan, I guess do it. But whatever we're working on, do it like like this, like, yeah, go through that sample test that you did, see which ones you got wrong, and note the topics that you need to review on, and then you can do content review for those topics.
Okay, and then you know how I said I don't really know like the behavior science.
Oh yeah, so I guess that's going to be like the necessary like content review stuff, which I guess like Kaplan will teach you like about, but I guess you're going to, yeah, I guess you're going to have to do some like content review for that. Like what I did was whatever I got wrong on, you know, any section, any like psych-social section, I would like make like an Anki card about it for that.
Well, I've been using Anki. I feel like I'm not using it. Like, I feel like I'm not making like great cards for it. But I don't know. I feel like I'm just using like I'm making the cards kind of weird. Where I feel like I'm making them over like things that are like really small details.
I mean, like, you know, the details add up, don't they?
Yeah, so I guess should I just do like more definition based for that?
Yeah, a lot of it is like just definitions, but like I would always just like kind of go by whatever it is I get wrong, or if I got it right by guessing, it still kind of counts as wrong because there's still much to learn there. You know what I mean?
Yeah, and that's like my issue is like I didn't like the only psych I took was like a psychology of like apes or something. So like I didn't learn like any of this.
Yeah. Me neither. Because there's psychology on it. There's sociology on it as well. So that was mostly like the new stuff that I had to learn for this test. So, but it's not like really really tough. Like that's why lots of people like are able to do well on it because they can, you know, like learn it because it's mostly like just content. They're not gonna you know throw any curveballs at you or anything like that.
Yeah. I mean I feel like a lot of like the words are also kind of helpful.
Yeah. For like PS, like I would just like basically like if I didn't know the definition of one of the choices, even if I got the answer right, I would still look up that definition.
Yeah, because I know I'm having like a really hard time with like all the brain.
Like anything in like particular, like
just remembering everything in the brain. I have like all parts for it, but it's weird because like the Kaplan video for it says find different definitions and functions than the Kaplan book, and then the back of the Kaplan book says like another thing. So I'm like, well, I don't even know what it does.
Well, I'm sure if you were asked questions on it, you would have like a better idea about it. Like, because I don't think they'll ask you anything specific like the laterate geniculate nucleus or something like that, which has to do with like the visual pathway, but like you know, no like limbic system, I guess, no like whatever the like
is like diencephalon like what comes from that important or
I mean if you know that like it's not part of the mesencephalon or the telencephalon, which are like behind and middle and then fore kind of like forebrain is like comes from the diencephalon. Yeah, I've never really saw a question that specifically asked you that, but
yeah, I guess that's because it's just
if you
I
can,
feel like I don't know how to sort out what I need to know versus what is like kind of because these books go in like way too much detail.
Yeah.
And, like, okay, so I don't know. Do you think I should even use the discrete questions in these books then?
Well, okay. I mean, if you like, first of all, do you have to like for your class and stuff like that?
No, I technically don't have to do anything for this class. It's like really weird. I think now that like it's online, it's not on Zoom, like it's on Adobe Connect, so nobody ever sees or hears me.
Yeah.
Like you can't even like turn on your camera or turn on your microphone. It's like very like corporate now. I feel like there's probably 100 kids in my class. So you're not really ever interacting with the teacher. They kind of just like talk the whole time
Yeah.
and do all the questions for you. That's I don't find it helpful at all.
Yeah. I think. Yeah, I think what's best is just to be able to do as much passage-based questions as possible that are as close to the AAMC test as possible. So, like...
Yeah, I feel like there's not a lot of... So all the questions, I mean, we do some of them in... All the ones, like, I do the pre-work for all the classes because I feel like that, then I kind of understand... Like, I don't have to do anything, but you get more out of it if you do it before because you kind of know what you're talking about. But all of the questions that I've done for Kaplan, literally every single one of them are discrete questions. That's why I think I'm like struggling so much with the passage questions. During class, like he'll do some passage questions, but the way he does it is just kind of like weird.
Yeah, most of the, like, you know, such a tiny portion of the test is discrete, and what people find difficult is usually, you know, comprehending the passages because they're really dense. Yeah. So, as much passage-based questions as you can. Because that also leads to, like, if you don't do as much passage-based stuff and focus too much on discrete, it also leads to, like, that, like, fallacy of believing that it's just all about content review when it's not.
Yeah, that's why I was like really shocked when I took the first test because I'm so stressed out reading these books, feeling like I need to know all the details, but then like 15 questions on like the subject, there's no way. It's probably not even a question in every chapter.
Yeah. It's try to do as much as much passage based stuff as possible, and from there, like, learn content.
So is this AAMC section bank, is this passage based then?
Yeah, basically the section bank is, because the question packs are old. They're like from the old MCAT.
Oh.
So they're like, there's one on chemistry, there's one on physics, and that's too specific. But for the section bank, it's closer to the actual exam because there's a section bank for CP, question bank for BB, question bank for... Section bank for CP, section bank for BB, section bank for PS. And then for cars, you should use the question packs.
Okay, that's what I was going to ask you, because like I know a lot of people... I was having a lot of trouble with my study schedule, so I know like some friends have sent me theirs and stuff, and everybody says that they do cars every day. Like, what? Where am I supposed to be doing cars every day?
Yeah. There's basically, there's no real good source of cars material besides the AAMC one, and there's a lot of AAMC car stuff, but yeah, sometimes like I have like students who have done them all, and I'm like, you know, I don't really think it's a good idea to like redo them because things just get weird in your head. Like,
you remember things and stuff.
Yeah. It's like proactive interference or retroactive interference. It's like one of those two. But but yeah, since you know you're just starting, it's going to be very like good for you. So like you could do cars every day, but you don't have to. It's not like you have to do like a ton of passages. Like I guess just like intersperse them in your study work or whatever, or you can even make like a day where you focus on cars, but you basically just have like the most important thing I think is just to like, I get see it here. I'll just write it in like an email for you. One sec.
Yeah, because I saw like all your spreadsheets, but
oh yeah, that reminds me. So like when you study, start like a stopwatch. Like I just Google stopwatch.
Yeah.
and I start that, and if I like go to the bathroom or like use the phone or like take a break or anything like that, I pause the stopwatch, because a lot of people like say that they study like all day, but it's not really like all day. And so, you know, what I find is the best is to like use like a stopwatch, and like whenever you're done studying, like input that into like a spreadsheet or something, so you know exactly how much work you've done on that day.
Yeah. Like I've been turning off my phone and stuff. But and I have all day, which is kind of nice. I think I'm starting my research like in 2 weeks, but I have all day now, and I have like this office, which is pretty quiet for the most part. That's why but that's why during the week I can't do it till after, because usually there's someone like right there.
Mhm.
We leave till like 3:30. But so I have this space that's pretty quiet, and I have like all day to study. It's like I gotta figure out what the hell to do.
Yeah. I highly recommend the stopwatch thing, because it's like, I think it's like Parkinson's law or something like that. It's just like, as if you give yourself 8 hours to do a task, it's gonna just take 8 hours. Like, you know what I mean? So there's like, yeah, so you want to get an exact number so you can kind of like look at that. Like for me, I find it very motivating. Like I see like myself, like if I can study for like 6 hours one day and I record that, I can slowly see like maybe the next day it's like 6 hours and like 5 minutes, but it counts. Like it's progress. Like every little bit counts.
Yeah, no, I get that. I think that's helpful. Also, just like more aware of taking breaks and stuff. I mean, because I have been here, like I've been here for a while, but I don't know if I'm like productive the whole time I'm here.
Yeah.
So then I'll do that. But then my question is, if I'm doing this is like 300 questions, so if I just start by doing this, don't you think, like, how long would this take me to do?
You're talking about the cars question packs?
No, like you're telling me to do the section bank first, right?
Oh yeah.
So like, wouldn't I be done with that kind of soon if I like sat here and just worked all day, pretty much?
Oh well, like think of it like first, like not necessarily doing the section banks, but first go to what you did in that sample test.
Okay.
And look at the like what you need to learn from that. Like if you need to learn something, then look that stuff up and learn that stuff.
Okay, so yeah, that's like a lot of stuff. So
Yeah. So then, you know, by the time you do like the section bank for, let's say, CP, let's say like, you know, and like you can do that however you want. I don't think it's necessary to start doing them timed yet. That's usually something I reserve for like the last like month or two, because if you do it timed like right away, you're going to be rushed and you're going to get like, the worst thing I think is when people like do questions and if they get them wrong, they just look at the answer and then they convince themselves that they won't get it wrong the next time. But if you don't time yourself and you give yourself as much time as you can, which it seems like you did for that sample test, then you're able to fully get
timed.
Okay. Well, yeah. So for the section banks, don't worry about timing. Like, if it takes you an extraordinary amount of time to answer like one question, it's all right, because that means that if you got it right, you're able to get that type of question right. Like, it's in your, like, you have the ability to do that.
Okay. So
Where
you're
it's
saying,
not, yeah.
yeah, because I know the sample test was timed, and that was also another reason. Like I didn't even. You could see like the last one we did with the ST whatever had no highlights, because like I literally was like I didn't even really get to read it.
Yeah. And yeah, at this point, since timing isn't like your number one priority at this point, since you're taking it in September, I think it's like a waste of the material, because it means that if you didn't have time to try it out and you just like look at what you got wrong and then you like read what the right answer is, it's like you never got an actual chance to actually do it.
So
I like to reserve time, like, because timing, like, the endurance thing and, like, being able to do it timed, that comes later. It's like when you first start to, like, ride a bike, or I don't know, like, what kind of, like, analogy to use, but, like, when you first start something, you need to, like, give yourself as much time as it requires for you to actually figure it out, for you to actually, like, have your brain go in the right direction, which takes a lot of trial and error. But once your brain is able to actually figure something out, no one can really take that away from you, because you, like, literally figured it out on your own.
yeah.
Whereas, like, if it was timed or something and you don't get a chance to figure it out on your own, it's almost like a waste of an opportunity for your brain to, like, get that kind of exercise.
Yeah, no, I get what you're saying, but so I get I should go through that and then just spend as much time as I need reviewing those concepts you're saying, like the ones I got wrong. Like there's no
Yeah.
spend as much as I need. But then also, so Kaplan gives us like 30. I do like about 30 something. It's anywhere from like 27 to like 40 questions before each class, like to just test what we know, and it'll be on like 4 or 5. No, yeah, like about 5 high yield topics, or chapters, or yeah, they're all like high yield science. So if I have not been doing well on those. So should I be also going through those, or like treating it as like not really that important?
Since the Kaplan material isn't as close of a representation of the AAMC stuff, or what's actually on the MCAT, I would just do it because I have to for the class. And if you still have any lingering questions from any Kaplan material, you can, like, certainly share that with me, and I can try to help you.
Yeah, but like if there's like a concept I don't get, should I just still study it the same way as like I am with the AAMCs for those questions? Okay.
Yeah. Just to, I guess, like, notice the difference between, like, it being discrete and it being passage-based. Because the passage-based stuff is by far the most important.
Yeah, and that's like what I'm having trouble with.
Yeah. Because there's only, like 12 discrete? Maybe a little more than that?
Yeah, around there. But then do you recommend UWorld?
That's also a good choice. I mean, that's also a good resource. It's just that they don't really give you that much. Like, they give you like a couple of months to use the material. If you are able to use anything else, I recommend like Blueprint, which is also known as Next Step, just because they just have harder. And again, since there's no other better source for CARS work besides AAMC, what I actually would do is, like, for the Blueprint exams, is just to skip the CARS section and just do like the CP, the BB, and the PS sections. But yeah, those are going to be like harder than what you see on the MCAT. And those, like, for instance, like when I did my spreadsheet on people who scored in the 100th percentile, like they had like an average MCAT score of like 524, and like roughly that range for the AAMC tests. But for the Next Step tests, the Blueprint tests, their average was like a 511.
Okay. Oh yeah, no, I did see that.
Yeah. And that's just because they're just like harder. So yeah. So I think that would be a good thing to think about. Because and also like UWorld is a great resource, and I would recommend it, but not as much as I would recommend like AMC followed by Next Step, because those things are like, not just like much closer to the actual exam, but like in the practice test format. Whereas UWorld, it's like kind of like, it's still like passage based, but it's like you can choose like the topic and stuff like that.
Oh, okay.
So, but it's not like, like practice test 1, practice test 2, practice test 3, etc.
But so I just feel like when I go through it, I'm gonna have to wind up like reviewing all of these topics and like every single chapter anyway. I just feel like there's something from each chapter that I don't know. So I feel like I'm still gonna spend so much time with content review.
I mean, like when we work together on like a passage, I'm going to like ask you like content stuff as we go through it. Like I asked you about like point mutations and other types of things.
Okay.
But it just the most I just want to like say like the most important thing is being able to deal with the passages.
Yeah. I definitely noticed that.
Yeah, because it's a passage based test, so you know, and content review. Like, do the content review as necessary based on how you do from the passage-based questions.
Okay, so I guess I'll re-go through my test.
Yeah. I think that's the best thing to do is to, go over the sample test that you took, go through, like, every question you got wrong, and try to, like, explain to yourself why the right answer is right and why the wrong answers are wrong. I think it's much more effective to like spend a lot of time. I think I mentioned this before, like a lot of time on a smaller amount of questions than to just take like hundreds of, or just do like hundreds and hundreds of questions and then like look at the answer and be like, okay, I get it. Because it's like, I would say go through the sample test, look at the questions, and the passages, and whatever you got wrong or you didn't understand, try to like understand them, and if you don't, you can send them to me, and we can go like that.
Yeah, so I'll do that, and then how do I go through the cars though?
Like how go, like what do you mean, like review it?
Like how do I review my cars? Because like
yeah.
Like I can't like go study a topic.
Yeah. Which is good because it means that you have to focus on like the passage as it is. But we can work on we can go over like cars like together like, next time.
Yeah, okay, perfect. Okay, I feel like that's it. Just like the spreadsheet I have to start, right? And then is there anything else I need to do?
I'll write like in an email to you like what to do like exactly. But it'll basically be like go over the sample test, figure out what type of content you need to brush up on. So, because I mean like instead of just like going at content like from the beginning of a book, like just as you do the passage based like sample tests and stuff, you'll find like holes, I guess, in your knowledge base, and then you can look up and shore up those holes, but based on the passages. Okay?
No, it's weird because I don't know. I feel like I'm like cheating by not like going through the books, but I'll go do that tomorrow then, starting tomorrow.
Because otherwise bio majors should score highest on the bio section, but they don't.
Yeah. I can believe that.
Yeah. So I think let me know when you want to meet next, and
how often like do you recommend meeting though?
I mean as much as you need.
Okay, so I guess I should wait obviously until I like review my whole test though to meet with you again.
Yeah, I mean it doesn't that like as review as much as you need. That could fill up like whatever.
Yeah, because I'll probably need all right, but then you don't think I need to push I'll need to push my test back? Do you think I have like enough time?
I mean, at this point, it seems like you have enough time.
Okay.
Because and you're gonna be working like on it like pretty much like every day, I suppose. But it seems like you have like 7, 8, 9. You have like 3 months.
Mm-hmm.
Or like 2 months and 20 days or whatever.
Yeah.
which is 2 2/3, right? Oh yeah, and I'll send you like the math stuff and stuff like that.
Yeah. Okay. So all that, and then the last thing is I have no idea what to make like a goal score. Should I just worry about that when I get closer?
Just think of the highest score possible.
Okay. Like I don't really like what's your goal.
Shoot for the shoot for the stars. You'll land in the clouds. At
Fair.
first.
Okay. All right. So how do I pay you? Do you have like Venmo or?
Yes.
Okay. Cool. And what do I owe you? And what is your Venmo?
So, yeah. Okay. Let me see.